All right. Thank you, everyone. We're going to begin with session 4 for our presentations today. And our first speaker is Edward Amory, coming from the Chevy Thorne Barton Arboretum in England. Edward has a background in politics, journalism, and business. And 7 years ago, he and his wife, Alice Thompson, who I also just met here at the conference, inherited Chevy Thorne Barton Arboretum from his father, Michael Heathcote Amory, an obsessional planter of oak trees from around the world. Michael was a member of the International Oak Society for several years before he passed and would have loved to be at a meeting like this full of fellow oak enthusiasts. But now Edward is going to share a bit more about his experience taking over the Arboretum and all things oak related collections and otherwise in England.
Thanks very much. And it's really exciting to be here and to meet you all. I'm just not convinced. Oh yes, here we go. No, we're getting not necessarily what I'm hoping for. I'm looking for the little pictures. Could some? Oh yeah, but before I get to the microphone, someone needs to fix my computer. I'm getting the notes coming up rather than the.
No, that's fine, but we don't see it.
Yes, you do, because I just pushed it.
Okay.
Yep.
But isn't that's the notes?
Oh yeah. Okay, fine. No, we're good. Okay. I'm here because my father was an oak obsessive. No, I'm going to. Sorry, I've got out of sync here. Okay, we'll start at the beginning. Most of this conference, as we've already heard today, is about the triumph of reason, careful taxonomy, scientific cultivation, propagation, leaf traits, landscape ecology, genetic DNA, foliar spectral signatures. But I want to talk instead about passion, obsession, emotion, and love. About how this passion has, over the centuries, motivated humans such as ourselves to collect, to cultivate, to share, and to explore the genus Quercus, and why that intense focus is needed now more than ever. I'm here because my father was an oak obsessive. When he died, Alan Coombs wrote memorializing him in your journal in these words: "How often do we hear of a remarkable garden or collection, only to discover that its inception and development is due to a single person? In my own experience, I've found such a situation to be very common, and the oak collection at Chevithorne is a good example, demonstrating that the most important factor in developing a good collection is passion, a force that never leaves us and drives us to achieve." I think that could apply equally to Alan as it can to my dad. Beatrice Chassé put it a different way. She talked about how a little bit of competition motivates us all. And as many of us know, it certainly motivates Beatrice. But call it what you will, this obsession led my father to create one of the great oak collections, of which more later. But my father was not alone. Passion and obsession have always driven oak collecting. There is a wonderful UK writer about trees, Thomas Pakenham, whose arboretum in Ireland is well worth a visit, even if oaks are not his main focus. His most recent book is about tree hunters of the 19th century and 18th century, and how they created the concept of the arboretum there. Here he is, last summer, showing us his Quercus afaris, well over 90 and still passionate about trees. Pakenham first takes us back to John Evelyn's book, Silver, published in 1664. This is what Evelyn had to say about planting trees, and not only were his words incredibly influential then, they still resonate today. It's what all persons who are owners of land may contribute to, and with infinite delight as well as profit, who are touched with that laudable ambition of imitating their illustrious ancestors and of worthily serving their generation. To these, my earnest and humble advice should be that at their very first coming to their estates, and as soon as they get children, they would seriously think of this work of propagation. Also, for I observe there is no part of husbandry which men more commonly fail in neglect, and have cause to repent of, that they did not begin planting betimes. Without which they can expect neither fruit, ornament, or delight from their labours. Men seldom plant trees till they begin to be wise, that is, till they grow old, and find by experience the prudence or necessity of it. Evelyn went on to urge landowners in Britain to plant what were not then yet called Arboreta, and many took him up on the challenge. In particular, they planted oak trees, and Evelyn, quoting a Jesuit poet, René Rapin, explained why. When ships for bloody combat we prepare, oak affords plank and arms our men of war, maintains our fires, makes ploughs to till the ground, for use no timber like the oak is found. Evelyn, and the arboretum he inspired, was important in another way. His work popularised new trees that thrived in the British landscape, replacing the destruction of forests in the Civil War that had just taken place. One of these newly arrived species was the tulip tree, of which we have a wonderful example in our garden in Devon. That's a picture. If I had to choose any favourite tree other than the oak, this would be it. And such plantings of new species, new arrivals then and now, provided resilience for Britain's forests against changes in climate, against diseases and pests, against change, because then and now, trees that arrived and thrived in an arboretum could end up being planted out across the landscape. This was the new era, and it bred a new breed of individual collector. One of the first of these, Henry Compton, was Bishop of London for 30 years, starting in 1675. He may not have been a passionate Christian, but what he really cared about was trees. As bishop, he was responsible for what were then called the American colonies, and he used his ecclesiastical network to collect new species there and bring them to the UK, where he planted them in the grounds of his palace in Fulham. Among the bishop's imports were Magnolia virginia, the first of the genus to cross the Atlantic, and the first UK example of Quercus coccinea, Virginia scarlet oak. Of course, like all collectors of trees, the bishop died too soon. His collection was dispersed, but he started a trend inspired by individual passion, but a trend that eventually had an enormous impact on the British landscape. The next great arboretum to be planted in Britain arose from an extraordinary collaboration between a young Quaker from Pennsylvania, John Bartram, incidentally the founder with Ben Franklin of the American Philosophical Society, and Lord Robert Petre, who inherited a great fortune and several huge estates when still a teenager. With Bartram's help, he began to import huge quantities of new plants and trees from America. He built the Great Stove, at the time the largest hothouse in the world, at 30 feet high, to propagate delicate species, and in 2 years before his tragic death at 29, planted 60,000 trees of 50 different species at his Thornton Hall estate. His passion and money made collecting trees hugely fashionable in Britain. He was also the first man to grow camellias in Europe. I'm going to give you one more example. In the 19th century, a British writer, John Loudon, first popularised the name Arboretum. Loudon published a successful magazine, the Gardeners magazine, the first of its kind, which brought the idea of the Arboretum to a far wider public in Britain and beyond. He also published a book, Arboretum and Fruticum Britannicum. He inspired others to share his passion, and the most extravagant and successful example of this was the extraordinary new Arboretum at Chatsworth, a collaboration between the vastly rich Duke of Devonshire, his young gardener and friend Joseph Paxton. It included 2,000 species of trees, many of which were extremely rare, and it began a new scientific approach to tree collecting. Each species of trees was laid out as an individual species plotted on a map, and it was open to the public, the first all year round, first of those sort of great estates to do that. So with only 20 minutes and 10 minutes left of your time, I'm now going to jump forward nearly 2 centuries to the house in Devon that my father inherited in the Devon countryside. It was passed down to him by my great-grandmother, a passionate gardener who lost her husband in the First World War and all 3 of her sons in the Second. She dealt with her grief and loneliness by planting, by creating what the gardening writer Lanning Roper described as a garden that had a controlled abandon that makes the garden romantic, informal, completely satisfying, and above all else unique. May, was her name, May found shelter and solace from the unendurable in her attempt to improve on nature. She took some fields and orchards, and through hard work, love and commitment, created one of Britain's great gardens. And when I knew her in her 90s, she was still on a kneeler in the flower bed. My father, that's my father with My father didn't share May's focus on plants. The house he inherited seemed like an expensive luxury. In fact, it took him years before he discovered his way to connect to the Devon countryside. But then he began to collect oaks, first a few and then many more. He suffered for the last 20 years of his life from Parkinson's, but his obsession with oaks kept him alive and active longer than any of us had any right to expect. The writer and neurologist Oliver Sacks has written about the impact of nature on his patients. He says, I cannot say exactly how nature exerts its calming and organizing effects on our brains, but I have seen in my patients the restorative and healing powers of nature and gardens. My father was a really good example of this. In planting oak trees, many oak trees, he found healing. He found friends, many of whom are here today, but he also found a passion, a vision of the future. Modern phrase which I like to describe something as a cathedral project. It's based on the idea that the early builders of cathedrals in Europe knew when they started they would never see the conclusion of their work. They knew they would die before it was finished, but they went ahead anyway, investing their lives in a vision of the future. A tree collection, an arboretum, is the same. My father wrote of his oaks. The next generation can decide what to do with them. They're not like daffodils. When they've been in the ground for a few years, they can look after themselves. Now my wife Alice and I are lucky enough to manage the legacy of that obsession. A collection of 220 species, 398 taxa, sort of give or take. 300 specimen trees include wonderful Quercus querquefolia, Quercus leucotrichophora, which I can't pronounce, and the autumn colors there in our garden of Quercus rubra from America. This extraordinary collection, the result of individual passion. Now it's up to us to find a role for the future. So what should be our mission as a relatively small private arboretum? First, resilience against pests and diseases. As a boy, we had an avenue of elms running up to our house in Devon. They all died in a single year of what we call Dutch elm disease. I look at the limes, and there you can see them, that my father planted to replace them, and yes, they are wonderful, but also a reminder of what we've lost and how quickly we can lose it. Now in Britain, we are losing our ash trees from ash dieback. I borrowed this graph from Simon Toomer. It shows the accelerating outbreaks of threats to trees in the UK. Climate change, obviously, is the main one. This is another Q graphic showing what will happen to their collection. The red dots are the ones most threatened. And the graph on the right is 2090. And you're showing that a whole bunch of the Q collection is threatened by this. And this shows the distribution of Quercus robur in terms of rainfall and temperature. You can see that by 2090, in terms of current projection, much of the world's population of Quercus robur is under threat. So we need to decide now what we're going to do about that. And we've started to look at how to focus for that. Trees for the future, our arboretum. Which trees from around the world, from all the countries that you come from, are going to thrive in Britain if Quercus robur declines? A third role for collections like ours in a relatively temperate climatic zone, stable political climate, is to provide a repository for globally threatened species. And we have many examples of species that are very vulnerable in their native habitat. We're putting together with the Hilliers, with Kew, with the Savile Gardens, we're instituting a UK meta collection on the basis that the best way to keep a plant is to give it away. Private collections have an advantage here, where we're a bit less worried if a species isn't very exciting. The only question is how to copy the trees, and here we're trying air layering. And finally, in Britain, like most advanced countries, we're becoming an urban nation, losing touch with our natural roots. There are lots of books about trees, but also a huge amount of ignorance. So we need to do more to encourage people to learn about our tree heritage. Our arboretum opens for about 30 days a year, a lot less than most public arboreta, but a lot more than most private tree collections. So we're trying to play our role in helping to educate the British public about why Quercus really matters. I guess that's where I've got to. I sped up a bit because I'm getting these little messages in front. And I will take any questions. But I suppose the thing that I would like like to take away from this is the sense that this group of people here, we're hearing a lot of science, some of it genuinely inspiring, but behind that science, behind all the graphs and the taxonomy, it's about passion, it's about emotion, it's about love. That's what we're all doing here, and that's why I'm very glad to be here in Oaxaca this week. Thank you.
Thank you so much for that presentation, Edward. Does anyone have any questions? Roderick?
So much of a question, but on your previous slide, there was a quote from John Evelyn that you didn't read, which I started to read. It looked really interesting.
Well, I was thinking, because in Britain, we've only got 2 native species of oaks. And so John Evelyn, this is, 500 years ago. So it is a pity that the several kinds of oak are so rarely known amongst us. And whenever they meet with Quercus, they take it promiscuously for our common oak. If anyone hasn't read Silver, I really recommend it. It's a great book. Yeah?
Question over here.
When you were a little boy, did you resist this passion?
Yeah, my dad used to have these oak weekends, including a number of people who may be here today, and I always used to try and hide. and yes, I can still remember Alan and others standing around an oak for about 45 minutes, arguing about which one it was. It's growing on me.
I see 3 questions.
Well, for me, it's a great privilege to hear you. I remember your father in the last Oak conference that he was present in Bordeaux. And I remember visiting your Cheviton Barton, excellent arboretum and a passion also. But one thing nowadays is the difficulty with this question of the Nagoya Protocol, with the difficulty of obtaining acorns from other parts of the world. I admire in UK, where all these botanists from so long ago were bringing in plants and nobody was complaining about invading something. And thanks to your father, that collection survives and you're here today. But for all of us that are in this country with all these magnificent collections, we look at them, but we know it's quite risky to put it in our pockets and take it away. Thank you very much.
My father, like a number of other people in the room, was a bit of a bandit.
Beatrice, did you have a question?
So my question is a sort of how do you say it in English? A good question to have after Peter's comment. So you talked about education and pests and diseases and the meta collection and everything. But what is your vision of the future of Chevy Thornbarton as an arboretum in terms of what you would like to plant still and where you see this arboretum going as an arboretum without any of those other considerations? Do you see what I mean?
I do. I think it's about finding your passion, isn't it? I mean, my great grandmother found the passion in plants to deal with this unimaginable loss of her whole family in the war. My father found a passion in oaks that took him through having Parkinson's for 20 years. And Alice and I and our family, we're finding a passion in integrating those 2 visions, the plants woman's garden, the English landscape that we've inherited, and the oaks from around the world that have just turned up, and integrating them into a single coherent whole. So that's our vision, if you like.
Do you have words of wisdom for those of us who carry on projects that were started a long time ago? So for not those cathedral initiators, but those that inherited a partially finished cathedral.
Yes, like me, absolutely. Well, I guess my answer to Beatrice's question is for me the answer to that. You have to find your way to become passionate about it. I'm not my dad. I can't tell you the name of every single oak in our collection. I can't. I would struggle to spend 45 minutes with Alan observing, working out which oak is which. But I'm here today because I found my own passion and Alice's own passion in oak trees, and I think it's about the people as much as the trees, to be honest.
I think we'll do one more question.
Oh, congratulations for your presentation, Edward. The pictures I took with your father and just Alan had many above. And the photo with your dad.
Oh, yes.
Yeah. I remember I took that picture. But it's not that my question. My question is that,
Oh.
yeah. My question is because your father was so proud about the Mexican collection, and I would like to know if you have noticed an advantage of the Mexican oaks growing in your property with the change in temperature that you mentioned.
Well,
Have you noticed anything?
It's a good question. And the answer is that the Mexican oaks are loving it. They're growing ridiculously fast. If anything, they grow too fast. If I talk to people, foresters, they worry that some of the Mexican oaks grow so fast in the British climate as it is now, that they will not be as useful as forestry trees. But they look really happy.
Okay, thank you.
All right. Thank you again, Edward, for your presentation. Okay, I was told that our next presentation is virtual. We'll be hearing from Dr. Philip Crim from the State University of New York, or SUNY, in Cobleskill, United States. Philip is a plant ecologist, founding director of Lime Ledge Botanical Garden and Arboretum in Elbridge, New York, and an assistant professor of plant science at SUNY Cobleskill. A lifelong tree enthusiast and woody plant obsessive, he founded Lime Ledge to promote biodiversity-based horticulture and connect his upstate New York community with conservation through education and public engagement. His work emphasizes native and edible species, ex situ conservation, and sustainable landscape design grounded in ecological research. And Philip is going to tell us more about the oak collection at his garden now.
Hi, my name is Philip Crim, and I'm going to be presenting today on building an oak collection based on local provenance, microhabitats, and conservation design. So I'm an assistant professor of plant science at SUNY Cobleskill in upstate New York, and we're going to be focused on how I'm using Quercus and a wide variety of oaks at Lime Ledge Botanical Garden and Arboretum. So, Lime Ledge, some quick facts, is located in Elbridge, New York, which is in the Eastern Finger Lakes region. So right about here, like right about in the middle of the state. It's very close to the city of Syracuse, which looks like this little, you can see the development on the map is this little kind of white area right here. We're about 20 minutes from downtown Syracuse. So Syracuse is sort of a mid-sized city in upstate New York. The metro area, I believe, is probably about just shy of 500,000 people. So it's not a very big city, but it's not tiny either. And the main road leading to Lime Ledge does get some pretty heavy traffic. So it's pretty convenient, pretty easy to get to from a fairly populated area. Lime Ledge itself, as an organization, was founded in 2023 They're a nonprofit 501c3 tax-exempt organization. Specifically, they are chartered with the State Department of Education with the state of New York. They are an environmental education organization, and they were founded to eventually purchase the property, which they did in 2024, to restore it. And this is a really rustic, basically abandoned farm property that comes with a lot of issues that similar farm properties have in the US, where there's piles of tires, there's old abandoned machines, etc. So we've been working on cleaning that up since we purchased the property in 2024. The site itself is about 227 acres, or about 92 hectares. We've got 100 acres in field crops and about 5 miles of trails that are in really good condition. So our trail system also has about 2 to 3 miles of additional trails that, with a little bit of investment and volunteer work, we could probably add to our existing trail system. We're right next to a county park, which also is a fish hatchery that has a few events every year and is pretty heavily trafficked. So our site is pretty well known, right from the start. We're right next to a pretty popular county park. We have about 15 acres that will be devoted to our collections right from the start as well. And I'll show you where those are on the next slide. We have a USDA Zone 6A climate, although the last 3 winters have been pretty mild. Last winter was the harshest winter in a few years and only went down to about -8 degrees Fahrenheit. So the 2 previous winters had been Zone 7 into actually like Zone 8 winters. The 2 years ago, that winter, the low temperature was only, I believe, I think it was like 8 degrees Fahrenheit. So it might have even been a little bit warmer than that. So by design, Lime Ledge is meant to be a living laboratory to test what plants can grow in our area. And that string of mild winters might have been giving us a little bit of a deceptive impression of what we can and can't grow there. But what makes Lime Ledge really special is that it has a wide range of topography and soil types. And we'll look at that in a few slides. So why do we care so much about oaks? Because it's such a large, diverse genus that we want to really fill all of these microhabitats at Lime Ledge with as many different types of plants as possible. And oaks offer us a really unique opportunity, because it's such a large and diverse genus, to really have one genus concept where there's so much variation in form and function that we can really use it for education in a way that most other genera just don't serve those same types of purposes. Oaks also are a large group that almost every person that I know, certainly, I won't speak about people on other continents, but if you're in the northern hemisphere, it's one of the groups of plants, or woody plants at least, that people probably interact with the most, or hear about the most in news and media. So there's this really important cultural component to looking at how humans interact with oaks and sort of using that as a hook for getting people interested. Unfortunately, one thing I've found is that there's a lack of public familiarity at the species level. A lot of people see the genus Quercus as this monolith that's really hard to begin to learn about and to discern the differences between closely related species. As somebody who teaches woody plant materials this semester at SUNY Cobleskill, a lot of my students come into that course with that impression, and we slowly introduce more oak species as each week goes by, until we have a week like just recently where we introduced like 4 in 1 week. And it kind of took us a few weeks to build up to that, just introducing 1 or 2 per week with a group of other species, until we start to drop more species to become familiar and build up that level of familiarity. If you're in Eastern North America and you haven't traveled very well, a lot of the oaks that you've probably seen are probably section Quercus or section lobate oaks. A lot of students don't have experience with anything other than those. So when they learn Quercus acutissima sawtooth oak, like they did this past week, it's kind of mind-blowing to see, for many of them, to see something that's neither in the white nor red oak groups. So very cool. A number of species also are narrow endemics. So from a conservation standpoint, being able to protect and retain biodiversity, it's really, really important to have that as a consideration in any oak collection. And indeed, in some collections, that might be one of the things that's most important about oak collections. But we're not just interested in conserving, necessarily species that are imperiled like narrow endemics, but also conserving local genetics, local germplasm. This is something that Lime Ledge wants to really focus on as well. So here's a map of Lime Ledge. You can see we've got on the northern side of the property, we are bounded by New York State Route 321, which is a pretty major road leading from the city of Syracuse into the Finger Lakes. Right next door on our western boundary, we've got Carpenter's Brook Fish Hatchery, that county park that I mentioned earlier. And then we have a whole range of habitats. Our trail map, our trails are all in blue right here. And we have a few overlooks. So Lime Ledge is on a prevailing north-facing slope. And even though the land is all like there's knolls, like for example, there's a knoll right here. There's a knoll here in the cook lot, like in our planting area. The land is extremely varied when it comes to topography, but the overall trend is a north-facing slope. And that helps give us a little bit more protection in the winter than some other sites nearby have. So, if we look at soil pH, and this, we really want to make a shout out to historical glaciation as well here, because this glaciation has really resulted in some fine spatial scale heterogeneity in the soils, where we can have soils with really different characteristics within literally a few meters of one another. So the soil pH is a good example of one of these characteristics that can vary somewhat across the site. In some places, it approaches 5, but there's enough organic matter in the soil that there's been so many areas that have been really well grazed or historically forested. There are no areas that are really erosive and exposing lower horizons. So most of the soils at Lime Ledge have a pretty high organic matter content. So even the areas that are pretty sandy and sharply draining still have pretty good water retention. But some of those areas are also fairly acidic as well. And then by contrast, on the other end of the spectrum, we have some areas like right here, kind of east and west of the farm quarry, which is this spot right here that's visible on the map. Fairly shallow rocky limestone soils where the pH can approach 8. But in most places, the soils are weathered enough that it's pretty much neutral. Like even right over the limestone in a lot of places, the pH does not become very alkaline. So this results in a really nutrient-rich medium for growing a lot of different species. We can grow Quercus muhlenbergii, chinkapin oak right near Quercus montana, aka Quercus pinus, within, again, a few meters of one another, because we have in the cook lot right here, where we have a lot of our oak species, the eastern end of the cook lot is primarily acidic, and the western end is a lot more dominated by clays and by higher, like the limestone's a little closer to the surface. The pH gets quite a bit higher, actually right along this boundary here. Things start to change pretty readily. So this western end of the cook lot, close to neutral. It's not really an alkaline soil. In a few places, the pH might go a little bit above 7. But we grow a few plants that prefer acidic soils on this side of the line, and they tend to do okay. We haven't had any major chlorosis issues. But something like a pin oak, Quercus palustris, we would definitely want to grow over. There's actually a sandy knoll that you can kind of make out here on the map. We grow a lot of conifers and some other acid-loving species, like, for example, Hamamelidaceae species in this area right here. And that's where we also grow some of our acid-loving oaks. But the really cool thing about the cook lot is that it allows us to grow taxa that have basically diametrically opposed soil preferences right near one another. So This area here that just got highlighted, this along our western property boundary is basically like a sandy ridge that goes south all the way back onto the rest of the county property here, well over our property line. And that sandy ridge, the pH is right around 5 in some places. It becomes quite acidic. It's also pretty sharply draining. You can't see it on the map, but there's a pretty substantial slope here on the western side of the property leading down to the fish hatchery. And the fish hatchery is called Carpenter's Brook because there's this stream that you can kind of see it meandering through the woods here. And it goes, that's what the blue line here is. And it goes all the way through here. And so this area right just to the west of our property boundary, this is a historical floodplain. And the soils are pretty coarse, but they're also pretty nutrient rich as well. And every once in a while, it does still flood. So this area on our northern corner is underwater sometimes for a couple weeks out of the year, but that's pretty rare now. This other area is our main planting area. So this includes the Cook lot right here, a second lot just to the north of it, which is dominated by a gravelly slope that goes down as you move west. And then on the other side of this, this hedgerow right here, this part of the lot, which we've actually, we've taken this off of the farmer's hands. This area here, we are reserving for species of Crataegus hawthorns. So we would like to have a substantial hawthorn collection at Lime Ledge for similar reasons as to why we are interested in having a large, diverse Quercus collection. So a little bit more detail on the soils. Our soils are dominated by inceptisols and entisols. And the entisols, especially the gravelly and steep terrain, this is more around like the southern part of the property in the woodlands, areas that it never really was economical to till and clear, and that's why most of the farmland is on the northern part of the property, and that's where we see a lot of our alpha sols, which are the productive valleys, foot slope soils. And that's also where our plant collections are as well. Most of our planting areas, our dedicated planting areas rather, are kind of on that interface right there. Although we scatter plants across the trails throughout the property in different nooks and crannies, depending on what's open and what's appropriate giving the site. One of our big philosophies at Lime Ledge is we want the land to basically guide what we do instead of vice versa. And one of the features of that I pointed out earlier, that farm quarry is one of the more special parts of this this mosaic of different landscapes that we have at Lime Ledge. We've tried a few different southwestern desert species here in the quarry, not just oaks, but a few other species as well, such as Buddleia racemosa, which we've been able to get through its first winter, and it flowered this year. So we're hopeful that we can get it to survive long term on some of these shallow spoil piles that have really shallow soil, which is mostly just rock shards, honestly, right here, limestone based. So fairly alkaline in some places, but again, even the limestone shards, like a lot of the soil is so heavily weathered because we're in a pretty high precipitation area that it's not super alkaline. So it's base rich, in other words, nutrient rich, without having a lot of severe alkalinity issues. If we're trying to grow a species that's perfectly happy in a xeric area, for example, different types of oaks from the Southwest that are happy on limestone, they are generally pretty happy here in our farm quarry. And this goes back to our microhabitat strategy. We want to use the farm quarry as a sheltered, well-drained site for some species. We're also experimenting with some alpine species that we're trying to grow near the tops of some of these, where the soil is well-drained enough, but they also get some shelter when it gets really hot, for example. So here, real quick, our hierarchy for the oak collection itself. Level 1 are Onondaga County natives, Onondaga County being Lime Ledge's home county in Elbridge. Level 2, the New York State oaks, so maybe some species that don't necessarily grow in Onondaga County, but might be nearby or other places in the state of New York, which is actually quite large. New York State's a lot bigger than a lot of people realize. And then North American species, which of course will emphasize We're some of the southwestern oaks for the farm quarry, also species of concern like Quercus bointanii, the Langtree oak as well. And then lastly, Eurasian taxa, the oaks that can be from basically anywhere on Earth, you know, that's obviously not tropical, because we're not going to have any luck with those plants in USDA Zone 6A. So section Cyclobalanopsis, for example, there's probably 2 or 3, I think, that we can grow for sure. And the rest of them we probably, you know, we wouldn't have any luck with. And even with those 2 and 3, we'd have to be very, very careful about how we cite them. Plants like Quercus mercinifolia, for example. So what do we want to do? We want to have our oak collection be a living reference for regional biodiversity, but also an ex situ gene bank for also species that might be like Quercus bointanii that really need some additional publicity and some additional attention. So what we really want to do is we want to position Limeledge within some existing well-recognized frameworks in order to collaborate with different collections and to be part of a network that's all working towards some of these same goals. We also want to make sure that we're getting our oaks ethically. We don't want to just go around and collect, you know, willy-nilly. We want to collect native oaks as much as possible from places where landowners are going to share with us. So they can be representative of that local germplasm, but also having partnerships with institutions, nurseries, and wild collectors that are doing things ethically, legally, and the right way. Obviously, some species, you know, there's not much we can do in terms of provenance. Quercus bointanii and the Langtree oak, which this is our Langtree oak in our collection here. You know, it's such a narrow endemic that it only grows in a couple sites, if it's even still extant in the wild, that we can't really be too picky about its provenance, because there really just is no diversity to choose from when it comes to that. But we've been able to keep it alive for 2 winters outside so far. So fingers crossed that it continues to succeed for us. Looking forward, our vision for Limeledge, we just got a big grant to renovate our barns. We have 2 barns on site to turn them into a environmental conservation and community center. So we see ourselves as a living gene bank, educational resource, and conservation hub, granting public accessibility with trails, interpretation, community-rooted programming, but putting some of these plants in front of people so that they can get a better appreciation for them. And so that genera like Quercus don't just look like a monolith that's impossible to tackle. You know, let's show them what the native ones are and generate and build their interest from there and start to show them why these species have been so important historically. So we want to be working with iOS and other institutions and other networks to really build our collections up and to be part of a broader community. We also just partnered this year with a native plant nursery, and that's the greenhouse that's being constructed right here near our barns. And they're planting this whole area with herbaceous species, which is what they specialize in. But fingers crossed, I've been pushing them to get a little bit more involved with maybe trying a few species of oaks. So thank you very much. Any questions? This is the stream on our property. I'll leave you with that. Thank you very much again.
Okay, thank you so much, Philip. It looks like there's at least one question out there.
Can you hear me?
Yes.
As a humorous comment, I'll mention that I wouldn't be too hard on the general public for their inability to identify oaks to species. Having just returned from the pre-conference tour looking at oaks in Mexico, the identification of oak species proved challenging even to iOS members. And more seriously, did I spy an image of World Begonia orchid in your presentation? Does that grow on your site, and is that associated with oak woodlands?
So I put that picture up because that's the cover page of the flora for Cortland and Onondaga County, which I use as a guide for finding sites for native oak species. For example, some of the oaks that grow in Onondaga County that we'd like to collect, like Quercus velutina, only occur on a few sites in that county. So we want to make sure that if we get some local germplasm to preserve that local provenance, that we can actually get it from as close to our site, like truly as possible. So there actually is a historical location, a historical spotting of small world Pogonia, not too far away, just outside of Syracuse. But unfortunately, it hasn't been spotted in several years.
Thank you.
Roderick?
Yeah, Phil, this is Roderick Cameron. Great to see you. I just want to say, really sorry that you weren't able to travel at last minute, but very thankful that this amazing technology can allow you to answer questions live. Still remember your great presentation in Las Cruces. My question is, fantastic what you're doing in Lime Ledge. I'm wondering if you're already at this stage thinking about succession. I mean, I don't know what the situation is with property rights, or what do you see 50 years down the line? Is there going to be trouble? Do you already thinking about that?
Well, the cool thing about Lime Ledge is it's owned by a nonprofit organization with a board of directors. So our objective is right now I'm the founding director, but I actually have a grant application out that would fund a full-time director whose main job would be going for that. So fingers crossed, I should be hearing about that later this month. But our long-term goal is to basically have like some permanent staff that are actually paid to have that as their primary job instead of just being a volunteer only organization like we are now as we kind of get on our feet. So long term, I'm hoping that Lime Ledge will be able to recruit some really top-notch staff members and hopefully a really, really talented and skilled director once we really get up and running.
Great, thank you so much. I think we'll wrap up our questions there.
Thank you so much, everyone.
All right, next in this session, we're going to hear from Dr. Tamara Citlali Ochoa Álvarez. She's coming to us from the Universidad Michoacana de San Nicolás de Hidalgo in Mexico. Tamara is a biologist who earned her Master of Science degree in integrative ecology. Her project focused on the leaves of the oak Quercus glaucoides, where she evaluated the chemical and nutritional content of its leaves and its relationship with herbivory and environmental variables. Tamara has a PhD in biological sciences, and her doctoral research focused on the response of Quercus castanea to drought, analyzing phenological and chemical variations in oak, as well as their interactions with herbivores. So now Tamara is going to speak more about this research on Quercus castanea.
Bueno, el trabajo que yo les voy a presentar es parte de mi tesis de doctorado, es el segundo capítulo. Y bueno, la herbivoría es una de las interacciones antagónicas más importantes que se da dentro de los ecosistemas. La herbivoría se define como tal, como el daño causado sobre cualquier órgano vegetal, ya sea dado por vertebrados o pequeños invertebrados. Tiene gran relevancia dentro de los ecosistemas, ya que participa en la estructuración de comunidades, así como en la regulación de los tamaños poblacionales de las plantas y en procesos de regeneración de los bosques. Varios estudios han mostrado que existe una relación causal entre el daño causado por los herbívoros y la disponibilidad de recursos de las plantas. la hipótesis de disponibilidad de recursos de Coley nos hablan que diferentes especies e incluso poblaciones de una misma especie pueden presentar distintos niveles de recursos y por lo tanto van a responder con distintas estrategias ecológicas. Por un lado tenemos las poblaciones que existen en hábitats donde hay bajo nivel, donde existe un bajo nivel de recursos, estas poblaciones van a adquirir estrategias ecológicas más conservadoras de recursos y por lo tanto van a ser poblaciones más tolerantes al estrés. Por otro lado, tenemos las poblaciones que existen en hábitats donde hay más, donde no hay una limitación de recursos y estas poblaciones van a adquirir estrategias ecológicas más de adquisición de recursos y van a tener mayor capacidad de crecimiento. También hay la relación que existe entre los insectos herbívoros y las plantas, pues puede estar significativamente afectada por el clima del área donde se encuentran. Aquí la temperatura y la precipitación juegan un papel importante, tanto en la diversidad de especies, en la abundancia relativa, así como en la actividad conductual de los insectos herbívoros. Y por otro lado tenemos que afecta directamente a la planta, teniendo implicaciones en el crecimiento, en la calidad nutricional, en la fenología foliar, así como en los compuestos de defensa. Todas estas características van a jugar positivamente o negativamente en la planta para desarrollar la resistencia a la herbivoría. El daño causado por los herbívoros también puede estar determinado por características propias de las plantas. Cómo los herbívoros toman en cuenta el estado nutricional de la planta, es decir, la cantidad de nutrientes que puedan encontrar en las hojas y también la cantidad de recursos que las plantas destinen a la defensa de la herbivoría. Aquí pueden producir una serie de metabolitos secundarios que van a funcionar contra la herbivoría, haciendo que la digestibilidad de la planta se reduzca. También cuando no participan en funciones de defensa, estos metabolitos pueden tener funciones de protección de la planta. Y los metabolitos secundarios, pues más estudiados dentro de los encinos, son los fenoles, específicamente los taninos condensados e hidrolizables. Un estudio que realizamos en el 2024, quisimos determinar cómo era la variación de la fenología foliar primaveral de Quercus castanea, dada por la estacionalidad de la precipitación. Para esto nosotros determinamos que la disponibilidad de agua que tuvieran nuestras poblaciones iban a jugar un papel importante en los tiempos de desarrollo foliares. Aquí, para poder determinar esta disponibilidad de agua, nosotros medimos tanto la precipitación como la humedad del suelo. Posteriormente, también analizamos algunas características foliares de los árboles para ver si estas variaciones que encontrábamos en la fenología foliar también afectaban otros caracteres foliares de los árboles. Para poder determinar la fenología foliar, nosotros tomamos dos variables. Una de ellas fue el tiempo de desarrollo. Esta se refiere al número de días que transcurren entre la etapa en la que los árboles se encuentran completamente sin hojas hasta que alcanzan el 100% de su desarrollo. Por otro lado, tenemos el E de 50. Esta variable se refiere al número de días que transcurren entre el inicio del año y hasta que los árboles alcanzan el 50% de su desarrollo foliar. Como resultados principales de este estudio, tenemos que la disponibilidad de agua sí jugó un papel importante en la fenología foliar. Y vemos que la humedad del suelo a principios de la primavera, que fue en marzo, tuvo una influencia tanto sobre el tiempo de desarrollo como con el ED50. Aquí todas nuestras poblaciones son diferentes figuras. Nosotros tuvimos 6 poblaciones y estos son todos nuestros individuos. Entonces, a mayor humedad acumulada en el suelo en el mes de marzo, vemos tiempos de desarrollo y ED50 más cortos, es decir, que tuvieron un crecimiento más rápido con respecto a las que no tenían tanta cantidad de agua acumulada en el suelo. También vemos que el grosor de las hojas se relacionó positivamente con el tiempo de desarrollo. Hojas más gruesas tuvieron tiempos de desarrollo más largos y por lo tanto crecimiento más lento. Entonces, tanto la fenología foliar como el grosor fueron diferentes en todas las poblaciones. Esto mostrando que existen mosaicos espaciotemporales de variación genética y ambiental y que estos pueden covariar con algunos caracteres funcionales de las hojas. Es por ello que para este trabajo nuestra hipótesis fue la siguiente. La disponibilidad de agua como factor ambiental va a tener una influencia directa sobre los niveles de herbivoría de Quercus castanea en la cuenca de Cuitzeo. Por otro lado, esta disponibilidad va a afectar características propias de las hojas, quienes pueden actuar como mediadores de este factor ambiental, sugiriendo que tanto el factor biótico como abiótico van a influenciar la intensidad de la herbivoría. Entonces, nuestros objetivos fueron evaluar una serie de rasgos foliares que influenciaran los niveles de herbivoría observados en Quercus castanea y posteriormente determinar el rol de los factores ambientales en esta herbivoría observada. Para ello, nosotros tomamos 6 poblaciones de Quercus castanea que están dentro de un gradiente de disponibilidad de agua basada en la precipitación anual de las poblaciones, que van desde 900 milímetros hasta 1300 milímetros de precipitación anual. Estas poblaciones se encuentran dentro de la cuenca de Cuitzeo, en el estado de Michoacán, en México. Quercus castanea es una especie de encino brevidecidua que tiene árboles que pueden alcanzar hasta los 18 metros de altura. Es muy importante ya que es una especie de encino que se caracteriza por estar en diferentes tipos de hábitats, que van desde bosques tropicales secos hasta bosques de pino encino. También cabe resaltar que es la especie de encino más abundante en nuestro sistema de estudio. Nosotros realizamos 2 colectas para el año 2021 y para el año 2022. Estas colectas de hojas se realizaron en el mes de diciembre, que es el periodo antes de que los árboles comiencen a tirar sus hojas y hacer el recambio. Con estas colectas nosotros determinamos los niveles de herbivoría, estimando primero el área total de las hojas y posteriormente el área dañada. También determinamos una serie de rasgos foliares, los cuales fueron el peso, el grosor foliar, el área foliar, el porcentaje de agua en las hojas, así como la fenología. También hicimos la cuantificación de algunos metabolitos secundarios y también de algunos nutrientes. Para el suelo nosotros medimos el porcentaje de humedad del suelo de cada sitio de los meses de primavera, ya que vimos en el artículo anterior pues que este era un determinante para la variación de Quercus castanea. Y también tomamos muestras de suelo de cada sitio para hacer análisis de capacidad de retención de agua. Nosotros tomamos la precipitación anual de la base de datos de World Crim y también tomamos la precipitación interpolada de datos que provinieron de estaciones meteorológicas de Conagua que estaban cercanas a nuestros sitios. Nosotros clasificamos tanto las variables foliares como nuestras variables ambientales. Las variables foliares las clasificamos de acuerdo al mecanismo que podría explicar la herbivoría. Entonces, las clasificamos en 5 mecanismos que corresponden a la cantidad de recurso, esto quiere decir la cantidad de recurso disponible para los insectos, a las variables que respondían a la defensa química, las variables que respondían a la defensa física, a la calidad nutricional, así como a la fenología. De igual manera, las variables ambientales las clasificamos en 3 condiciones de disponibilidad de agua distinta. Y aquí fueron las variables que corresponden al gradiente de precipitación. Tenemos la precipitación de Workley y la precipitación interpolada. También variables que responden a la precipitación reciente. Esta es la precipitación a los meses previos de las colectas, y también las variables que responden a o que caracterizan más bien la humedad de agua en el suelo. Para poder establecer o para poder responder si nuestra hipótesis cuáles eran los factores ambientales que podían tener un efecto sobre los procesos que controlan la herbivoría, nosotros realizamos modelos de ecuaciones estructurales. Primero, realizamos 2 hipótesis generales donde establecimos que las variables ambientales, esas son las 3 condiciones en las que las clasificamos, iban a tener un efecto sobre los rasgos foliares de Quercus castanea, y posteriormente este efecto se iba a ver reflejado en la intensidad de la herbivoría. Por otro lado, tenemos la siguiente hipótesis, que es que la disponibilidad de agua iba a tener un efecto directo sobre la herbivoría sin necesariamente haber afectado los rasgos que pueden influir en esta. Para no hacer uso de todas las variables que nosotros medimos, quisimos seleccionar los mejores indicadores de cada uno de tanto de los mecanismos como de las condiciones. Para ello, realizamos una serie de modelos generalizados mixtos, y con ello pudimos hacer una selección de los mejores indicadores utilizando un criterio de información arcaica. Para los rasgos foliares, escogimos uno de cada uno de los 5 mecanismos. Aquí nos quedamos únicamente con el área foliar, la concentración de fenoles, la relación nitrógeno-fósforo, el porcentaje de agua contenido en las hojas, así como el tiempo de desarrollo. Por otro lado, los mejores predictores de la disponibilidad de agua fueron la precipitación anual interpolada, la precipitación del mes previo a la colecta y la capacidad de retención de agua en el suelo. Finalmente, este fue nuestro resultado, este fue nuestro modelo de ecuaciones estructurales, donde nos muestran cuáles fueron las correlaciones significativas que existieron entre nuestras variables. Los cuadros azules muestran las variables de disponibilidad de agua, los recuadros verdes muestran los rasgos foliares, y el recuadro amarillo muestra la herbivoría, que es el porcentaje de área foliar consumida. Las flechas de colores son las correlaciones significativas que nos arrojó el modelo. Las que vemos en color azul son correlaciones positivas y en color rojo son correlaciones negativas. Las flechas de un color uniforme son las correlaciones que hay entre la disponibilidad de agua con el porcentaje de área foliar consumida a través de los rasgos foliares. Y las flechas punteadas son las variables de disponibilidad de agua que tuvieron una correlación directa con la herbivoría sin necesariamente haber afectado algún rasgo foliar. Bueno, primero tenemos que la precipitación anual interpolada se correlacionó positivamente con el área foliar y con la concentración de fenoles, aunque únicamente la concentración de fenoles en las hojas influyó directamente sobre el porcentaje de herbivoría. Vemos que las poblaciones que tuvieron una mayor precipitación anual tuvieron una menor concentración de fenoles en sus hojas, y a menor concentración de fenoles en sus hojas fueron menormente defendidas y por lo tanto tuvieron más incidencia de ataque por herbívoros. Por otro lado, vemos que la precipitación del mes previo a la colecta se correlacionó positivamente con el porcentaje de agua en las hojas y negativamente con la relación nitrógeno-fósforo, aunque solo el porcentaje de agua en los tejidos tuvo una influencia directa sobre el área consumida. Entonces vemos que a mayor precipitación del mes previo a la colecta, los árboles acumularon mayor cantidad de agua en sus tejidos y hojas más suculentas mostraron mayor incidencia de daño por herbívoros. Y finalmente vemos que la capacidad de retención de agua en el suelo se correlacionó positivamente con el área consumida. Es decir, que sitios que tenían mayor capacidad de acumular agua fueron también mayormente atractivos hacia los herbívoros y por lo tanto hubo mayor incidencia de daño. Eso quiere decir que los insectos herbívoros sí están tomando en cuenta la disponibilidad de recursos, en este caso del recurso hídrico, para poder atacar ciertos parches que ven atractivos hacia ellos. Como conclusiones, tenemos que los porcentajes de herbivoría que encontramos en Quercus castanea fueron determinados principalmente por la concentración de fenoles en las hojas. A menores concentraciones de este metabolito secundario, pues mostraron mayor ataque. Y el contenido de agua en las hojas también fue un factor que influyó fuertemente. Las hojas más suculentas mostraron mayores niveles de ataque. Tanto la precipitación anual como la precipitación del mes previo de la colecta también tuvieron una influencia significativa sobre el daño que vimos de los herbívoros. Sin embargo, esta influencia fue indirecta, ya que influyeron a través de la modificación de ciertos rasgos foliares, como lo fue la concentración de fenoles y el porcentaje de agua, que fueron los rasgos foliares que estuvieron directamente relacionados con la herbivoría. Y finalmente, el único factor ambiental que se correlacionó directamente con la herbivoría fue la capacidad de retención de agua en el suelo. Individuos que tuvieron mayor capacidad fueron los más susceptibles al daño. Gracias.
Muchas gracias a la doctora Tamara. ¿Alguien tiene pregunta? Any questions?
Gracias. Gracias por la presentación. Dos preguntas. La variable que indicas como retención de agua es lo que también es conocido como la capacidad de campo, medida. Ok. Ahora, lo que me parece curioso es que no hubieran una relación directa entre la cantidad de nutrientes, nitrógeno, fósforo, con la herbivoría, porque uno pensaría que es a lo que están enfocados los bichos, ¿no? Por la palatabilidad. Sin embargo, se ve que hay una disyuntiva entre qué tantos nutrientes hay y qué tantas defensas hay, porque sí hay correlación con los taninos. Entonces, pues, no sé, esta disyuntiva, ¿crees que sería algo solamente de la especie o del tipo de herbívoros particulares para la especie, o se podría inferir hacia la sección o demás encinos del área?
Bueno, creo que esto bueno, yo lo he visto en más especies de encinos, que como tú lo mencionas, o sea, sí toman en cuenta como la cantidad de nutrientes, pero también están muy enfocados como a las formas que tienen las hojas, bueno, que tienen los árboles de defenderse. Entonces sí, como que su referente mucho, aparte de las defensas químicas, también son las defensas físicas. Entonces también hojas más gruesas y más duras, o sea, hace que impida que haya mayor incidencia de herbívoros. Entonces, sí se van, al menos yo lo he visto en encinos, como por el lado de ver cómo están defendidas bueno, la parte que van a consumir, más que tanto como la cantidad de nutrientes. Pero yo creo que eso sí pues puede variar y también depende mucho, o sea, como de las especies de los insectos herbívoros.
Gracias. Mi español está malo, pero also, in English, what was the average rate of herbivory that you saw throughout the study? Were the herbivores eating a large percentage of the leaves or a small percentage of the leaves? And do you know much about what herbivores were there in your study system?
Bueno, mira, el porcentaje de herbivoría en las hojas, aproximadamente en todas las poblaciones fue pues relativamente bajo, ¿no? O sea, se manejó como entre un 30 y un 40% del área total de las hojas. O sea, sí. Y digamos, los herbívoros que más vimos en Quercus castanea, fueron pues lepidópteros, bueno, larvas de lepidópteros, sí.
Muchas gracias.
I think we can do one more question if anyone has one. All right. Gracias, Tamara. We'll wrap up. Our next speaker is Goretty Nayeli Mendoza Juárez. She is here from the Instituto de Investigaciones en Ecosistemas y Sustentabilidad de la UNAM en Morelia, Mexico. Goretty is a PhD student of the Biological Sciences program at UNAM. Her research has focused on exploring coexistence patterns through functional and climatic niche analysis tools in oak species from temperate forests. Recently, her interests have expanded to investigating foliar endophytic fungal communities associated with oaks, recognizing their ecological relevance and the complex interactions between host and microbial communities. So now Goretty will speak to us more about this research.
Bueno, pues buenas tardes a todos. Este es un... Hola. Gracias. Buenas tardes a todos. Pues es un gusto para mí estar aquí hoy con ustedes. Mi nombre es Goretty Mendoza y les voy a presentar, a platicar un poquito sobre este proyecto titulado Alta diferenciación en las comunidades de hongos endófitos en encinos que coexisten tanto rojos como blancos. Entonces, bueno, a manera de introducción... perdón. Bueno, a manera de introducción, los hongos endófitos foliares pues son aquellos, o los podemos definir, como hongos que viven parte de su ciclo de vida en tejidos de las plantas como las hojas, sin causar síntomas visibles. Entonces pertenecen principalmente al filo Mascomicota y bueno, los podemos encontrar a lo largo de un continuo de interacciones que bueno, además del mutualismo o la patogénesis, también los podemos encontrar como comensales o como saprófitos, cuando viene la senescencia de las hojas. Ahora bien, esta interacción que se ha dado a lo largo de millones de años entre microorganismos y sus hospederos, pues tiene ciertas implicaciones. Entonces, esto lo que puede afectar o estas alteraciones están involucradas, por ejemplo, en las tasas tanto de fotosíntesis como de crecimiento del hospedero, pero algo muy interesante es sobre todo su papel que tienen como en la defensa de los, en contra de organismos patógenos y herbívoros. Bueno, en cuanto a la estructuración de estas comunidades de hongos, pues bueno, aquí tenemos o podemos ver un corte transversal de una hoja, en donde para que estos organismos puedan colonizar el interior de la hoja, pues existen diferentes factores implicados. Entonces, por ejemplo, a una escala regional, tenemos factores abióticos, como por ejemplo, pues la precipitación, la altitud y el clima. Ahora bien, a una escala más local, pues están implicadas aquí las interacciones bióticas, pero también el microclima. Entonces imagínense justo pues esta hoja, pues a las condiciones a las que está expuesta. Entonces, por ejemplo, aquí tiene que ver, por ejemplo, la incidencia de la luz UV, también tiene que ver la humedad o pues cuestiones como del microclima, que está dado también por rasgos funcionales. Ahora, estos rasgos funcionales pueden servir como barreras y están relacionados a aspectos de la planta tanto morfológicos, por ejemplo, aquí tenemos el rasgo, por ejemplo, el área foliar específica, fisiológicos, como es la cantidad de clorofila o el pH, y de nutrición, como pues la cantidad de nutrientes, entonces puede ser carbono, nitrógeno o fósforo, ¿no? Como los principales. Ahora bien, existen estimaciones de más de 2.2 millones de especies de hongos endófitos. Sin embargo, pues bueno, como se imaginarán, la gran mayoría pues no están descritos. Estas estimaciones, pues bueno, se sabe que estos hongos pueden presentar alternancia entre órganos, por ejemplo, pues las raíces, tallos y hojas. También se ha visto alternancia entre flores, frutos, semillas, también entre los tejidos del xilema o del floema y entre diferentes hospederos. Y bueno, también algo que habíamos mencionado previamente es la alternancia al
alternance to.
Bueno, también algo que habíamos mencionado previamente es su capacidad de la alternancia al saprotrofismo durante la senescencia de las hojas. Okay. Entonces, bueno, pues surge aquí la pregunta de que si estos hongos son generalistas o especialistas. Entonces tenemos por un lado, pues artículos o trabajos como estos, donde nos menciona que la abundancia de las plantas, pero no la historia evolutiva, es la que moldea los patrones de la especificidad del hospedero en endófitos foliares. Entonces esto nos está hablando de que los endófitos prefieren colonizar hábitats pues que están disponibles y que son abundantes, más allá que pues irse por una historia evolutiva. Pero también tenemos a la contraparte, ¿no? Donde artículos como estos pues nos mencionan que los rasgos que pertenecen o que forman parte del espectro económico de la hoja están asociados con comunidades de hongos endófitos foliares. Entonces, pues bueno, quisimos explorar un poquito pues estos puntos de vista usando a los encinos como modelo de estudio. Entonces, pues bueno, algunos de los hospederos más conspicuos de estos hongos pues son justamente los encinos. Bueno, previamente se mencionaba que había alrededor de 161 especies registradas en el país. Esta mañana, pues vimos que no es así, que hay al menos 179. De estas, pues gran parte son especies endémicas y estos encinos, pues presentan un alto valor tanto económico como ecológico. Y bueno, para el estado de Michoacán, así como para muchos otros sitios, pues estos se encuentran distribuidos en gradientes de riqueza de especies, ya que, por ejemplo, este mapa pues nos muestra zonas donde el color verde más intenso son zonas donde hay hasta 26 especies potenciales y el color blanco pues pueden ser zonas donde no existen especies de encinos aquí registradas, bueno, potenciales, ¿no? Bueno, entonces lo que sabemos es que dentro del género Quercus, tanto la sección Quercus como la lobate, pues bueno, tuvieron una diversificación de manera paralela en Norteamérica y posteriormente irradiaron hacia México. Entonces, esto conllevó a la presencia de una evolución simpátrica en los mismos nichos, tanto climáticos como de suelo, y esto pues conlleva a patrones importantes de coexistencia entre ambas secciones. A partir de esto, pues bueno, tuvimos dos preguntas de investigación. La primera de ellas es que, bueno, si la composición en las comunidades de hongos endófitos foliares están asociados a la filogenia de los hospederos. Y la segunda pregunta es si la variación funcional entre estos clados de encino explican la composición de estas comunidades de hongos. Bueno, nuestro muestreo se llevó a cabo en el estado de Michoacán, dentro de la cuenca del lago de Cuitzeo, la cual, bueno, la elegimos debido a su alta heterogeneidad ambiental. Y lo que hicimos fue que colectamos hojas vivas a partir de 9 especies de encinos, como hospederos, y esto para 11 sitios dentro del estado de Michoacán, dentro de esta cuenca. Entonces, primeramente, pues medimos caracteres funcionales como la concentración de clorofila, el pH, el área foliar específica, el grosor foliar, la densidad y longitud estomática, el contenido de taninos, el contenido de agua, y también determinamos algunos nutrientes como el carbono, nitrógeno, fósforo, así como radios estequiométricos de estos, ¿no? El carbono, nitrógeno, carbono, fósforo y nitrógeno, fósforo. Y bueno, por otra parte, lo que hicimos con estas hojas colectadas fue que hicimos una extracción de DNA, tanto a partir de las muestras como a partir de cepas de hongos que nosotros aislamos a partir de pequeños pedacitos de hojas. Estos aislamientos que nosotros hicimos previamente, los utilizamos para conformar una mock community, para saber qué hongos nosotros teníamos para el procesamiento bioinformático posterior. Bueno, después de la extracción de ADN hicimos un PCR anidado y para esto utilizamos los primers ITS1-Cata e ITS4-NGS-UNI. Y elegimos estos ya que comprenden a la región de ITS completo, lo cual pues pasa por la región 5.8S. Estas muestras se enviaron a la Universidad de Wisconsin para una secuenciación a través de la plataforma de Pacific Biosciences. En cuanto al procesamiento bioinformático, bueno, con los archivos crudos, lo primero que hicimos fue una conversión a formato FASTQ, después una evaluación de la calidad. Elegimos este pipeline para hongos, el llamado Pipecraft, donde, involucra diferentes pasos. Entonces, tenemos, por ejemplo, el filtrado de calidad, después lo que hace es un clustering y finalmente hace una asignación taxonómica utilizando la base de datos de Unite. En cuanto al procesamiento, pues lo primero fue remover las secuencias de Quercus que había por ahí, así como las secuencias cortas también. Y también removimos las secuencias correspondientes a la mock community que previamente pues agregamos. Finalmente hicimos una asignación taxonómica, pero esta vez de manera manual. Esto fue utilizando el programa Genius y también utilizamos la base de datos de Unite, ya que pues es la base de datos curada para los hongos. Los plots pues los hicimos mediante el software R. Bueno, en cuanto a resultados, pues les muestro estas dos tablas donde pues aquí aparecen las especies de encinos donde se muestraron. De este lado aparecen para la sección lobate y del lado derecho para la sección Quercus. También podemos observar el número de muestras, el número de individuos muestreados dentro de cada especie, así como la riqueza de los otros obtenidos. Entonces podemos observar que tanto castanea como Quercus obtusata fueron las especies con mayor representación en cuanto a número de muestras. Y bueno, obtuvimos 119 otus en total, los cuales se distribuyeron en 9 especies de encinos, 47 individuos y 11 poblaciones. Ahora bien, a modo de análisis exploratorio, pues lo primero que hicimos fue graficar la frecuencia, distribución de los 119 otus obtenidos. Y lo que encontramos es que la gran mayoría de estos aparecían únicamente en una muestra. Es decir, esto es a lo que le llamamos los singletons. Había otros que aparecían únicamente en 2 muestras, o sea, los doubletons, y había otros pocos que aparecían en 3 o más muestras. Bueno, estas son las gráficas que obtuvimos para ver estos 119 otus, pues a qué grupos de hongos pertenecían. Y entonces encontramos que el 84.2% pertenecen al Phylum Ascomycota y el 17.6% al Phylum Basidiomycota. Y bueno, aquí les cuento que la diferencia entre estos dos filum es el modo de reproducción, ya que los ascomicota lo que presentan son estructuras, sus estructuras reproductivas son como unos sacos o unos globos, lo cual hace pues que las esporas salgan disparadas a presión. Entonces, por eso es que quizá la mayoría de los hongos endófitos foliares pues están en la parte aérea. Y por otro lado, los basidiomicota pues son hongos que están muy asociados al suelo. Y esto es porque su estructura o sus estructuras reproductivas son a modo de unos basidios o bastoncillos, los cuales pues bueno, se genera esta estructura y las esporas caen por gravedad al suelo. Entonces, esta es la diferencia entre estos dos grandes grupos. Entonces, bueno, esto nos está hablando de que tenemos sí hongos, pues en la parte aérea de las hojas, pero también hay hongos que, bueno, que nosotros los determinamos como endófitos foliares porque estamos aislándolos de hojas que están en la parte aérea, pero que en bases de datos pues aparecen como que son hongos del suelo. También, bueno, dentro del filum Mascomicota, pues las principales clases que encontramos fueron primero pues los dotiomicetos, que son este color como cafecito, y también, perdón, los dotidiomicetos y después los leotomicetos, que son estos de color naranja, dentro del filum Mascomicota. Y dentro del filum Basidiomicota, los tremelomicetes son los que predominan con un 81%. Bueno, aquí tenemos una gráfica de la presencia de las familias de hongos endófitos foliares que se encontraron. Entonces, bueno, del lado izquierdo tenemos las 4 especies de encinos muestreadas para la sección lobate y del lado derecho tenemos las 5 especies muestreadas para la sección Quercus. Entonces, bueno, primero, a modo general, lo que podemos observar es que para los encinos blancos, pues se ve una mayor riqueza de familias de hongos asociadas. Entonces existen algunas familias que no están, que no están en los lobate. Por ejemplo, tenemos la familia Didimelaceae, que aparece en color morado, que no existe o no se registró esta presencia en lobate. Y también la familia Didymospheriaceae, que es este color como entre verdecito, azulito, que tampoco se encuentra en lobate. Y bueno, también, o sea, sé que estos son nombres que quizá no pueden tener mucho sentido, pero les cuento que dentro de aquí también existen levaduras. Entonces, por ejemplo, existen géneros como Cándida, otro que se llama Mellerosima, Piquia, que son levaduras. Entonces, esto nos indica que existe la presencia de levaduras en árboles de encino, ¿no? Bueno, también otra cosa pues que me gustaría contarles sobre toda esta riqueza que vemos de familias es que si nosotros nos vamos a bases de datos, por ejemplo, una base de datos muy común para los hongos es una que se llama Fungal Traits y Fungal Traits contiene como estilos de vida tanto primarios, secundarios, terciarios para alrededor de 10,000 géneros de hongos. Entonces, pues bueno, buscando estas familias o bueno, los géneros que aparecen, pues resulta que estos hongos están anotados tanto como endófitos foliares, pero también aparecen como epífitos, pero también aparecen como saprófitos del suelo o aparecen como patógenos de plantas. Es decir, quizá tiene que ver pues con la alternancia que veíamos al principio, de que pues pueden alternar entre sus modos de vida. Este, y bueno, también pues algo que llama la atención es que para Quercus glaucoides, ay, para Quercus glaucoides, pues vemos una riqueza, una alta riqueza de familias de hongos, a pesar de que Quercus glaucoides solamente, o bueno, fue la especie que tuvo una menor representatividad en el muestreo. Entonces, pues aquí esto es algo que llama la atención. También, bueno, las barras grises son aquellas que no se logró una asignación taxonómica hasta la parte de familia, sino que la asignación taxonómica fue a un nivel taxonómico mayor, ¿no? Entonces, pues bueno, son grupos de hongos que todavía pues están inexplorados o no sabemos qué sean. Bueno, para el análisis filogenético, pues bueno, lo que se hizo fue este árbol mediante una aproximación de máxima verosimilitud a partir pues de las 119 secuencias fúngicas, las cuales fueron manualmente editadas y alineadas. Entonces, bueno, ¿qué tenemos? Pues que tenemos en la parte de arriba del árbol este color como rosita, son los hongos que pertenecen al filum Basidiomycota, mientras que estos color verde acá son los Ascomycota. Entonces, lo que podemos ver es que existen ciertos grupos de hongos que parece ser que tienen o que están asociados a cierto hospedero. Por ejemplo, si ustedes se fijan del lado derecho, pues hay ciertos colores. Entonces hay grupos de hongos que, por ejemplo, solamente están asociados a Quercus, que son el color morado, ¿no? Mientras que existen otros grupos o otros clados como este de aquí, que solamente está asociado al ovate. Y realmente son pocos los que están asociados a ambos, o sea que son los que están en color amarillo, ambas secciones. Y bueno, aquí pues el p-valor fue significativo, quiere decir que encontramos señal filogenética en este caso. Ahora bien, exploramos la parte desde los caracteres funcionales que medimos previamente y pues bueno, aquí obtuvimos índices como de Pagel y de Moran, en donde, bueno, encontramos señal filogenética para, bueno, el índice de Pagel es para encontrar señal filogenética a una escala mayor. Entonces lo encontramos tanto para el contenido de taninos como nitrógeno total, fósforo total, y relaciones estequiométricas como carbono-nitrógeno y carbono-fósforo. Y por otro lado, el índice de Moran es aquel que se utiliza, pero a una escala más local, para determinar más específicamente qué grupos de hongos pueden presentar señal filogenética para ciertos rasgos. Entonces, bueno, es lo que está aquí marcado en azul, quiere decir que para estos grupitos de hongos, pues existe señal filogenética para este rasgo, ¿no? O lo que está aquí marcado en azul, pues también este grupo de hongos, mismo caso. Y bueno, también se hicieron comparaciones pareadas de acuerdo a, a las especies más abundantes. En este caso, bueno, tenemos Quercus castanea y Quercus obtusata. Entonces, bueno, aquí determinamos señal filogenética para la densidad estomática. También, bueno, este es el caso de Quercus castanea con Quercus deserticola. Aquí, bueno, el p-valor fue marginalmente significativo. Y pues de nuevo aquí tenemos grupos de hongos, pues como asociados hacia este rasgo, ¿no? Que en este caso es el área foliar específica. También tenemos acá pues el nitrógeno total. Y bueno, nuestras conclusiones para este trabajo es que encontramos evidencia de que la composición en las comunidades de hongos endófitos, la composición de hongos endófitos foliares está asociado a la sección de los encinos como hospederos. También que bueno, que algunos rasgos funcionales foliares, como el contenido de taninos, nitrógeno total, fósforo total y radios estequiométricos carbono-nitrógeno y carbono-fósforo, influenciaron la composición filogenética en las comunidades de estos hongos. Y bueno, este trabajo resalta la importancia de rasgos evolutivos y funcionales en estos árboles como hospederos, en moldear las comunidades de hongos endófitos foliares en ecosistemas de bosques templados. Y bueno, en cuanto a agradecimientos, pues me gustaría agradecer al Laboratorio de Genética de la Conservación del IES de la UNAM, bueno, al Laboratorio Nacional de Síntesis Ecológica, al Instituto de Biología, pues por el desarrollo de este proyecto. También, bueno, a la CECITI por la beca otorgada durante los estudios de posgrado. Y bueno, a la doctora Tamara Ochoa por su apoyo en campo, así como, bueno, a Violeta Patiño y también al maestro en ciencias Diego Isla por su apoyo tanto en laboratorio como en la parte de bioinformática. Y sería todo. Muchas gracias.
Muchas gracias, Goretty. Any questions?
Gracias. Gracias por la presentación, muy interesante. A mí, acá estoy. Eh, a mí, bueno, una de las dudas que me surgen es, nos hablaste al principio como que de este salto que pueden dar los hongos endófitos a ser saprótrofos, ¿no? Y hay trabajos que sugieren que la descomposición de la hojarasca comienza incluso cuando todavía está la hoja en el árbol, ¿no? Entonces, con esta idea en mente, ¿qué crees que tendría más importancia en el futuro proceso de descomposición de estos tejidos? Ya vimos las diferencias entre las dos secciones. La sección Quercus tiene mayor cantidad de familias, mientras que la sección Lobate menos. ¿Qué crees que tendría más importancia? O sea, ¿habría un favorecimiento en el proceso de descomposición por una mayor cantidad de familias o menor, porque si no competirían entre ellas? O si tienes alguna idea de las familias que viste, qué tan fácil es que den este salto hacia la saprotrofia. Gracias.
Pues yo creo que es muy difícil determinar esta parte porque no se sabe con certeza en qué parte de este continuo de interacciones se encuentre. O sea, podría ser que estos hongos estén como organismos latentes que ni siquiera se encuentren en el proceso de saprotrofismo, ¿no? Pero quizá los encontremos mediante estas herramientas como presentes, pero que no estén ejerciendo o que no estén, pues sí, ejerciendo como un efecto como tal de degradar estas hojas. Entonces, pues sí, sí sería, yo creo que difícil determinar como esta parte, pero sí, efectivamente hay como un balance, ¿no? Entre las defensas de los árboles y lo que está ejerciendo el hongo como para poder eh, cambiar o estar en esta estrategia, ¿no? De ser como patógeno, comensal, saprótrico. Ujú.
Hola.
Um,
Muchas gracias por una charla muy interesante y me gusta tus resultados mucho. Pero yo tengo una pregunta. ¿Es posible que las diferencias entre las especies dentro de la sección se deben al muestro? Por ejemplo, ¿cuántos árboles por especie y cuántas hojas por árbol? Porque hay diferentes diferencias entre las especies, pero posible es biológico o posiblemente es estadístico. ¿Qué piensa?
Sí, yo creo que, bueno, aquí tuvimos una situación con el muestreo que al momento de hacer el PCR anidado, hubo unos situaciones técnicas donde al final los resultados que nos mandaron, pues hubo esta discrepancia o esta diferencia en el tamaño de muestreo. Entonces tenemos especies con muy poca representatividad y otras con muchísima representatividad. Yo creo que sí sería importante, pues en futuros trabajos, como intentar igualar esta N, como para ver si estos patrones se siguen manteniendo, o si hay diferencias. Pero sí creo que fue un problema más bien bueno, no del muestreo inicial, sino más bien de, bueno, los resultados que obtuvimos del secuenciador debido a este PCR anidado.
Gracias.
Pero muchas gracias.
Thank you, Goretty. Okay, next we'll hear from Doctora María Camila Latorre Cárdenas. She is also from the Instituto de Investigaciones en Ecosistemas y Sustentabilidad from UNAM in Morelia, Mexico. María Camila is a postdoctoral researcher at UNAM, and her research interests are in conservation biology and genetics, particularly on understanding the effects of landscape transformations and fragmentation on demographic, genetic, and ecological aspects of priority and threatened populations. Currently, her work centers on the understanding of functional connectivity, richness patterns, and microevolutionary processes in temperate forests of central Mexico. So now I'll pass it over to María Camila.
Ok, thank you. Voy a hablar en español. Bueno, buenas tardes a todos. Bueno, pues vengo a hablarles justamente de encinos, pero también en un contexto del hábitat de ellos, ¿no? El ecosistema de ellos, que son los bosques templados, en donde pues también comparten con otras especies, como son los pinos. Bueno, pues bueno, ya pues la mayoría de ustedes conocen los bosques templados de México. En esta imagen pues pueden ver la distribución que tiene este bioma. Cubre cerca del 17% del territorio de México. Y bueno, pues como ya sabemos, México es pues un centro de diversificación de los encinos, pero también de los pinos. Este ecosistema es muy importante porque pues provee muchos servicios ecosistémicos a los humanos y también a otras especies. Sin embargo, pues está bajo bastantes amenazas, pues principalmente la deforestación, ¿no? Que se debe al cambio de uso de suelo, al cambio en los regímenes de fuego y a la tala ilegal. Entonces, pues tenemos cerca de un 1% de una tasa anual de deforestación. Y en el estado de Michoacán, pues justo esta tasa de deforestación en algunas partes alcanza a tener tasas mucho mayores del 1%. Entonces, bueno, pues estas son imágenes de cómo es el bosque templado en Michoacán. Pues se ha reportado que el 20% de la pérdida del bosque dentro del periodo del 2001 al 2017 está asociado justamente a la expansión del aguacate. Entonces, bueno, pues ya saben que México es el primer exportador de aguacate y el 70% de todo lo que se exporta en México justamente proviene de Michoacán. Entonces, pues muchas veces cuando, pues muy probablemente todo el aguacate que consumen en otros países salieran de Michoacán. Entonces, bueno, pues es justamente como muy importante estos procesos, ¿no? De conversión de los bosques en Michoacán, principalmente por el aguacate, pero también hay otros cultivos como las berries que también influyen mucho, ¿no? En esta deforestación que tenemos allí. Y bueno, en esta imagen les muestro lo que se denomina el mosaico socioecológico de la franja aguacatera, es decir, en donde pues tenemos esta expansión de los cultivos de aguacate. En verde, este verde oscuro, ven todo lo que es el bosque templado. En naranja son todo lo que son los huertos de aguacate. Entonces, si pueden ver como que está justamente más, pues crece más, ¿no? En todo lo que es la parte occidental de este mosaico, aunque también aquí tenemos unas partes en la zona oriente. Ahora también quiero resaltar la presencia de 5 áreas naturales protegidas. Una es el pico de Tancítaro, que está aquí en la parte occidental y que si pueden ver está embebida, ¿no? Dentro de muchos huertos de aguacate. La otra es la reserva de la biosfera de la mariposa monarca. Es muy importante justamente porque llega esta especie. Y también tenemos otras 3 que son un poco más pequeñas, pero que son muy importantes porque hacen parte como de este corredor, ¿no? Del bosque. Ok. Entonces, pues justo dentro de este contexto de lo que son los paisajes socioecológicos, pues es muy importante entender que tenemos que encontrar un equilibrio entre el desarrollo humano, ¿no? Que pues justamente involucra todos estos procesos, ¿no? Agrícolas, ¿no? De expansión de producción, pero también encontrar un equilibrio en poder conservar la biodiversidad, ¿no? Y pues justamente para esto, pues en términos globales hay como muchas iniciativas. Una de ellas es la iniciativa 3030, que se busca en el año 2030 conservar el 30% del territorio, ¿no? Mundial, ¿no? Y entonces justo dentro de este contexto, pues una de las cosas importantes es identificar y priorizar pues cuáles son esas áreas importantes para la conservación, para poder conservar la mayor biodiversidad y justamente la integridad de los ecosistemas. Entonces, como parte de esto, pues el objetivo de este proyecto que les estoy mostrando es identificar unidades de conservación de Quercus castanea y priorizar áreas de bosque templado dentro de este ambiente socioecológico dominado por el cultivo de aguacate, teniendo en cuenta diferentes escenarios de cambio climático a futuro y también de cambios de uso de suelo. Y para esto usando herramientas genómicas y de ecología del paisaje. Entonces, bueno, pues muy rápidamente, pues nuestra metodología consistió en usar a Quercus castanea como pues, nuestra especie modelo. Esta especie tiene una distribución muy amplia en México y es representativa del bosque templado. Y lo que hicimos fue que colectamos en 23 sitios, 88 individuos. Hicimos como todo este proceso pues del laboratorio para obtener un set de 24.000 SNPs usando el método de secuenciación de GBS, Genotype by Sequencing. Y después de esto lo que hicimos fue filtrar como en dos grandes grupos, uno en el que contenían nuestros SNPs neutrales y otros nuestros SNPs adaptativos para justamente entender procesos neutrales que están asociados a la demografía de flujo génico y por otra parte entender procesos de adaptación local. Después de esto lo que hicimos fue pues conocer la estructuración genética, también evaluar patrones espaciales de la composición genómica actual y a futuro de esta especie, a futuro justamente usando diferentes escenarios de cambio climático. También estimamos diferentes patrones de conectividad del bosque en estos escenarios y finalmente como, combinamos todo eso para hacer una priorización pues de los bosques, ¿no? Entonces, bueno, ¿qué encontramos? En términos de la diversidad genética, cuando vemos esta set de datos neutrales, pues encontramos que tenemos solamente un grupo genético, es lo que vemos en, pues todas los puntitos son las poblaciones que mostramos. Bueno, no las poblaciones, los sitios. Y pues encontramos que nada más tenemos una población, un grupo genético, pues que comparte pues una historia demográfica y que mantiene un alto flujo génico. Y esto está soportado por estos valores de diversidad genética. La heterocigosis fue moderada, 0.26, tenemos una baja endogamia y una baja FST, lo que nos habla de que hay mucho flujo génico entre estas localidades. Sin embargo, en los análisis que hicimos de estructura genética teniendo en cuenta los SNPs adaptativos, bueno, potencialmente adaptativos, pues encontramos 4 grupos, ¿no? Entonces, de este que hemos encontrado neutral, se desprenden otros 3 que están como justamente en los extremos de la franja aguacatera, ¿sí? Entonces, esto es muy importante porque pues realmente es importante tratar de conservar todos estos pues procesos, ¿no? Adaptativos pues para poder asegurar la resiliencia de esta especie frente a pues todos los cambios que vamos a tener en el futuro. Ok, lo otro que hicimos fue justamente conocer la composición genómica actual de Quercus castanea. Y bueno, en estas imágenes lo que ven es, bueno, pues sí, la composición. Entonces, entre más diferentes son los colores, significa que en términos genómicos son más diferentes también las poblaciones que habitan en esta zona, ¿no? Esto es cuando usamos todo el set de datos y esto es cuando usamos nada más los SNPs adaptativos. Entonces, si se dan cuenta, pues hay un gradiente, ¿no? De adaptación local, porque las especies que encontramos en el occidente son muy distintas a las del oriente, al igual que también encontramos un gradiente de norte a sur en esta parte del este. Y esto se refleja más en este gráfico, en el cual las zonas que están más calientes son las zonas que tienen una mayor diferenciación, ¿sí? Entonces es donde las poblaciones tienen distintas adaptaciones. Y bueno, pues ¿cuáles son esas adaptaciones, no? Entonces, de los análisis que hicimos, pues justamente encontramos 288 SNPs, que son los 6 candidatos, ¿no? Pues que hace parte, ¿no? De estas adaptaciones locales. Y estas son las variables bioclimáticas con las que se asociaron, ¿no? Las más importantes son variables de precipitación. La bio 18 es la precipitación en el cuarto más cálido, así como la 19, que es la precipitación en el cuarto más frío, y esta que es la estacional, ¿qué? Se me fue la palabra, la estacionalidad de la temperatura. Y bueno, a partir de estas asociaciones, ¿no? O de estas posibles adaptaciones locales, lo que hicimos fue pues ver cuál es el genomic offset. Si recuerdan, en la mañana ya nos habían hablado otras charlas sobre el genomic offset de otras especies de Quercus insignis, ¿no? Y bueno, el genomic offset lo que nos habla es como la cantidad de cambio genómico que necesita una población para poderse ajustar a los cambios ambientales a futuro, ¿sí? Entonces, actualmente las especies están adaptadas a unas condiciones, pero en el futuro van a cambiar y no sabemos si van a poder hacer ese cambio pues y al mismo tiempo o igual de rápido que como están cambiando las condiciones, ¿no? Entonces, aquellas que necesitan un cambio mucho mucho más fuerte, pues van a ser más vulnerables porque puede que no alcancen, ¿no? A adaptarse, ¿no? En el futuro. Entonces aquí en este mapa, justamente las zonas que tienen colores más cálidos son aquellas en donde las poblaciones van a ser genómicamente más vulnerables. Entonces, bueno, esto está para diferentes escenarios, el 2030, 2050, pero en el 2070 es donde pues vemos, ¿no? Estos cambios más drásticos y bueno, que realmente son una preocupación, porque si vemos justamente las poblaciones que están dentro de las reservas, pues son las que van a estar más vulnerables. Y esto responde mucho a que en el futuro justamente las condiciones de precipitación van a disminuir mucho y también va a haber una alteración en la estacionalidad de la temperatura. Ok, bueno, lo otro que hicimos fue medir los patrones de conectividad y para esto consideramos dos escenarios, ¿no? Uno es la dispersión de las semillas, que en términos generales no alcanza más de 1 kilómetro, y el otro en términos del polen, que en general alcanza los 10 kilómetros, 20 kilómetros y en algunos casos hasta 80, ¿no? Pero normalmente pues alcanza 10, 20 kilómetros, ¿no? Para dispersarse, que esto es muy importante justamente para que se mantenga, ¿no? La conectividad del bosque. Entonces lo que hicimos fue que medimos o bueno, estimamos esta métrica del ECA, que lo que nos dice es la cantidad de superficie dentro de la franja aguacatera que está conectada, ¿no? Entonces, si vemos, pues a lo largo de los escenarios se predice pues que va a reducir, ¿no? Esta conectividad y obviamente el escenario en donde disminuye más, pues justamente es el de las semillas, pues porque son las que tienen una dispersión más limitada. Entonces, bueno, esto pues también nuevamente es importante porque casi se disminuye en un 25% la conectividad. Sin embargo, nosotros identificamos cuáles son los parches de bosque que son más importantes para mantener la conectividad, ¿sí? Y esto a través del índice compuesto de conectividad. Entonces, pues aquí les presento, ¿no? Como los diferentes parches que son importantes, prioritarios para conservar la conectividad. Y aquí les presento justamente cuáles son esos parches que son importantes para mantener la conectividad del bosque, tanto de la dispersión de las semillas como del polen. Entonces, bueno, después de haber tenido todo esto, lo que hicimos fue combinar estos datos y hacer una priorización en términos de cuáles son los parches más factibles de conservar y cuáles son los que van a ser un desafío. Los que son más fáciles de conservar son aquellos que son muy importantes y que tienen una vulnerabilidad genómica baja, ¿sí? O sea, en el futuro no van a estar pues afectados, ¿no? Por los cambios climáticos. Y la otra categoría son los que van a ser realmente un desafío porque son importantes, pero en términos de la vulnerabilidad genómica puede que los podamos perder en estas áreas. Entonces, en colores azules tenemos los que pues, son más fáciles de conservar y justamente están en la parte occidental, y los de la parte oriental son, pues, son los más vulnerables, ¿no? Em, y bueno, pues esto se puede traducir en que los que son factibles de conservar, pues lo que tenemos que hacer es como monitorear estas zonas, decretar algunos parches de bosque como áreas naturales. Y en las zonas que son difíciles, pues es probable que necesitemos estrategias de migración asistida, pues justamente, ¿no? Para ayudar a estas poblaciones a que pues puedan sortear, ¿no? Las condiciones futuras. Ahora, obviamente estoy hablando nada más de Quercus castanea dentro del contexto del bosque, ¿no? Pero obviamente hay muchísimas otras especies. Entonces, justamente en el laboratorio hay un estudiante, Vanessa Izaguirre, quien hizo este mismo trabajo, pero con una especie de pino, con Pinus leiophylla. Y pues lo que hicimos fue justamente pues, hacer una comparación de qué estaba pasando con él, y realmente sí comparten muchas áreas en las que pues hay una alta vulnerabilidad, ¿no? Sobre todo aquí en la parte del occidente, o estos parches, ¿no? Entonces esto nos va a ayudar a empezar pues como que a generar estrategias para poder priorizar la conservación. Mucho de esto también pues, puede estar soportado por la identificación de zonas de transferencia de semillas, que ya se ha hecho para otras especies de pino justamente en la zona, y pues coincide mucho, ¿no? con las zonas que nosotros estamos encontrando como vulnerables. Ahora, otra cosa para considerar, pues es justamente la expansión del aguacate. Hay algunos estudios que justamente han predicho que bueno, las partes que ven en morado son las partes en donde se va a seguir expandiendo el aguacate, ¿no? Entonces, no nada más las especies van a ser vulnerables frente al cambio climático, sino que también va a haber una pérdida, ¿no? Del hábitat, justamente por la expansión del aguacate. Entonces, pues va a ser muy importante eh, pues empezar como a entender un poco más cuál va a ser el papel que tienen las áreas naturales protegidas en esta zona, ¿no? Y bueno, manera de conclusión, pues bueno, pues los bosques de Michoacán, pues bueno, están enfrentando diferentes amenazas. Y bueno, se estima que la conectividad va a disminuir cerca de un 25%. Sin embargo, usando pues todas estas herramientas genómicas y ecológicas, pues pudimos identificar cuáles son los parches que nos pueden ayudar a mantener la conectividad, y que desde ahora eh, pues tenemos que enfocarnos en estos, ¿no? Para llevar a cabo diferentes estrategias de conservación que nos permitan mantenerlo. Sobre todo, pues en estas áreas que les mencionaba, ¿no? De la reserva de la biosfera de la mariposa monarca y en el pico de Tancítaro. Y bueno, pues agradecer. Les quiero comentar que, bueno, estas personas obviamente también contribuyeron muchísimo a este trabajo, por lo cual pues también les quiero agradecer ahorita. Aquí tienen... Ups. Oh, no cambiaron la presentación. Bueno, les quería mostrar un folleto que justamente estamos haciendo para la conservación de los bosques de Michoacán. Y les había puesto el QR, pero bueno, en otro momento se los puedo compartir. Y ya, muchísimas gracias.
Muchas gracias a ti, doctora María Camila. Does anyone have a question?
Thank you for your excellent presentation. My question is, are there any avocado companies that are ecologically ethical who could help you conserve the forest while also producing avocados that are sustainable?
Ok, ya. ¿Por qué está hablando en inglés? Sí hay o sea, bueno, hay de todo, ¿no? O sea, hay un gradiente, ¿no? O sea, hay productores que realmente sí usan, ¿no? O tienen como un tipo de producción, ¿no? Del aguacate un poco más amigable con el bosque. Y que estarían dispuestos, ¿no? Como justamente a ayudar a mantener ciertos parches de bosque, etcétera, ¿no? pero también está esta otra fase, ¿no? De que pues realmente hay productores que son muy grandes y pues realmente sus estrategias o sus estilos de producción pues no van tan alineados, ¿no? Con la conservación, ¿no? Como que hay todo este gradiente y pues sí, más bien como que aquí el reto es acercarse, ¿no? A este tipo pues de personas que sí tienen como esta iniciativa, ¿no? De, de tener como este tipo de producción un poco más amigable, pues como para empezar, ¿no? A hacer estrategias, digámoslo así, de conservación, ¿no? En términos estatales, pues ya se están implementando algunos, pues como algunas leyes, ¿no? Por ejemplo, digamos, está prohibido, o sea, ya está como, se habla como de la tala cero. Entonces, digamos que los aguacates o sea, bueno, los huertos de aguacate que se identifican que son productos de la deforestación, no se les permite, ¿no? Que puedan exportar, ¿no? Entonces, como una manera en la que pues se puede trabajar con ellos, ¿no? Pero sí, es todo un reto, la verdad. Sí, respondí tu pregunta.
Yes.
Ok.
Sabido que
We had one question over here.
Yeah, sorry, mine's on a sort of similar thing. I was going to ask what pesticides are being used for to control what kind of pathogens they may get, if they get fungi or if they get insects. So what kind of pesticides? Is it insecticides or fungicides? And these may have spray drift and they stay a long time in the environment. And what effect would they have on the oaks? And the ectomycorrhizal fungi and the mammals that may be around that may disperse the seeds.
Ok. No te entendí la primera parte, pero... Ajá. Ok. Pues bueno, específicamente, ¿qué tipo de pesticidas y eso? No sé, ¿no? O sea, sé que están de cierta manera regulados, pero aún así son muchos pesticidas los que se usan. Y claramente sí tienen un impacto, ¿no? Sobre el ambiente. Sobre los encinos realmente no sé si tienen un impacto. O sea, que yo sepa, no se ha estudiado todavía eso. Digamos, en términos de otros mamíferos y otros animales, sé que sí tienen un efecto, porque tengo un pasado, pero con otras especies de animales, en las que sí sé, ¿no? Que ese tipo de pesticidas que son muy volátiles permanecen mucho en el ambiente durante muchos años y realmente sí tienen repercusiones sobre el sistema endocrino, pues de los humanos y de otro tipo de animales, ¿no? Entonces, sí es un problema ambiental. Realmente no está como tal regulado. No se conoce como tal qué efecto tenga sobre los encinos, pero sí son cosas muy importantes, ¿no? De tener en cuenta y de trabajar.
Probablemente no tenemos más tiempo para preguntas, pero muchas gracias, María Camila. Un aplauso para ella. Next up, I think we'll be switching to English for the presentations. We're going to be hearing from Dr. Victoria Sork, who is at the University of California, Los Angeles in the United States. Victoria is a distinguished professor of ecology and evolutionary biology at UCLA, and also the director of the university's Mildred E. Mathias Botanical Garden and Herbarium. Victoria studies evolutionary and ecological processes in tree populations, especially oaks in multiple North American ecosystems. Her research combines landscape genomics, functional genomics, conservation genomics, and epigenetics with common garden and greenhouse experiments to understand adaptation and maladaptation to current and future climates. And currently, her research includes 8 California oak, mainland, and island oak species to determine how long-lived California oaks will survive rapid climate warming, which we'll hear more about now.
Good afternoon, and thank you very much. So I'm going to try to focus this talk first on establishing why there's a problem and then what we can do about it. And let's let's see. All right, I always like to start with the acknowledgements because so many people help me do this work and support it and produce all the wonderful data. So we have multiple funding sources, people from, 15 years ago that helped us get started, recent participants. I've had the pleasure of 2 postdocs from Mexico who've contributed along the way. And my Sork lab has just always come in and collected data. So I want to thank all of them and just remind all of you that my name's on it, but it barely represents the village. So the question is, are oaks adapted or maladapted to current climate conditions? So we think, and too often we think that the way we see things now, everyone's happy, everything is adapted. And I just want to point out that only 20,000 years ago, California had much cooler climates. And in terms of the generation time of a long-lived tree, that's not really that long ago. So if we look at the current, you can see by the gradient, the yellow is there's much parts of it that are much warmer than they were 20,000 years ago. Maybe it matters, maybe it doesn't. But if we look at what the differences are, what are the areas where oaks are located? So this is an outline of the valley oak distribution. But you can see there's areas that actually have had a lot of change in the last 20,000 years. And that may affect the extent to which oaks are adapted to where they live today. But what about oaks in future conditions? So current distribution, I just showed you that. If we look at a predicted using the 8.5 model, that's the one that's business as usual. We don't get any better at what we're doing. You see there's a lot of areas, not the same areas, by the way, as we saw from differences from the past. These are now going to be, the temperature is going to be different in the next 50 years, much shorter time than 20,000. And here again is the differences in just that one temperature variable. So I just want to point out, things change and oaks have to deal with it. And they've been really good at dealing with it. That's why we have so many species. They have been able to speciate and respond, but can they do it now? You've heard a lot about landscape genomics and adaptation. So I want to do is give you a cartoon so that everybody, I'm not going to show you any of my flows. I'm not going to show you how I do genomics. You're just going to believe that I know how to do it. But I want to show you, here's like a map of a landscape. And we can see there's an underlying gradient, different, environments on the left side of that figure and different. And if there's local adaptation, we're going to have trees that are adapted. We sort of indicate that with color to whatever the color is below them. And then trees that are adapted to, let's say that's a warmer climate on the right? So now we have these trees living there. Now we're going to change. What happens? Those trees, those were the trees 20,000 years ago. Now we've got post-glacial warming, and that's what it looks like. All of a sudden, those trees adapted to 20,000 years ago, if they haven't evolved, are now going to be maladapted to current conditions. And we're, assuming, well, in 20,000 years, won't they have evolved? So you can look at this and ask this question by looking at the phenotypes. Are phenotypes adapted to the last glacial maximum performing well in current climates? It's testable. Or you can do is look at the genotypes, and landscape genomics uses genetic variation to try to infer whether trees adapt to the last glacial maximum. Or we can take it with our paradigm with rapid climate change. You can see the top shows the same individuals, but maybe everything's gotten warmer, and all of a sudden you see, especially in some areas, there are individuals maladapted. We can measure this by looking at phenotypes, like how they're growing, how big they are, how they're reproducing, or we can use a landscape genomic approach, and we can use genetic information in the distribution. And you've just seen a lot of, and so the premise of landscape genomics is you can use genes to detect the degree to which adaptedness has changed. So the topics of mine are, I want to show you through our research evidence of maladaptation, but you've seen it in many talks today. So we've got lots of examples. Then I'm going to talk about, well, what could we manage our oak populations so that things get better? or is there anything to do about it? So I want to actually give you some solutions. And then finally, I'm going to talk about recommendations to preserve oaks and to preserve oak ecosystems. So let's just talk about some of the studies that we've done in my lab on evidence of maladaptation. I'm going to talk about valley oak in our long-term trials. So I'm going to actually look at the phenotypes. Then we're going to look at blue oak, which is a widely distributed oak in California, and do a landscape genomic analysis. So I'm going to switch to the genetics. Then I'm going to talk about California island oak, Channel Islands, and this is, we'll use landscape genomic. Now, this is just for the first part of the evidence. Most of this talk, I'm going to kind of take time on the evidence, and hopefully I don't get too long and can't get to the solutions. So here's a provenance study that we set up about 13 years ago. We intended to be at least 20 years. Two big, huge 2.5 hectare plots. We've got 95 provenances or localities or stands, but basically sites throughout. But the more important thing is that we have 672 families. And this gives us a lot of statistical power to look for genetic differences. And we're now in year 13. So what's the rationale for our design? We want the maximum number of families to test for genetic differentiation. So through 672 is pretty good. We want more trees, and we want them all over for climate association tests. So I don't deal with the population. I deal with where trees and families are located. It gives you a lot more statistical power and allows you to look at the landscape. And then we have 2 gardens. Be nice to have more, but it's a lot of work just to have 2 so that we can test for plasticity. So how can provenance test for local adaptation? So in a provenance, what you do is you've collected seeds from moms located all over the state. You would think if they're locally adapted to the climate of that garden, the ones who come from the nearest should grow better. And as you get further away, you get to either hotter areas or cooler areas, you're not going to grow as well. So that's what that little cartoon shows. You know, so we call that the transfer distance. What is the distance between where that lineage came from and the climate of the common garden? But there's other possibilities, as I keep alluding to. You could have lag adaptation. The lag would be, well, no, they haven't quite been able to track the temperature change in the last 20,000 years. So we have the local adaptation hypothesis and the transfer distances, and then you have the adaptational lag adaptation, which is no, they haven't kept quite track. That's an alternative. That's just talking about what are your populations looking like today. This is our result. That clouded area, that's the observed pattern. Now that dashed line is local adaptation. That's if we were locally adapted to the common gardens, they would be peaking. The best growth would be the ones that came from nearby climate. In fact, we don't even get the peak. In order to kind of get a sense of where would that peak be, we did a Gaussian estimate, okay? A Gaussian estimate, and then generated a new peak, and then we can look at the adaptational lag. So in Valley Oak, I was really surprised to find out that they're not even adapted to current climates, which exacerbates the problem of what are they going to do in the future. So Valley Oak is adapted to temperatures more similar to last glacial maximum, and how will these phenotypes survive in the future? So you can do is use those provenance trial information and look at to see what's going on. And just a quick summary, California is a heterogeneous mix of climates. So when we look at this map, the light spots are lineages that grew well in the common gardens and the dark aren't. So it's not like it's all from the south or we've got a clear gradient. It's really very mixed. Most trees are adapted to climates cooler than their current location, and the provenance studies identify which phenotypes will grow better. So this is really, but not wonderful, but not everyone has 20 years to get the data to figure out what to do. And that's why we're looking at landscape genomics as a possible alternative. So I'm going to now switch over to the Blue Oak study. This is a work we're doing in collaboration with the Nature Conservancy because they manage a lot of properties and they'd like to get more information. And So we're going to use genomic tools. I'm not going to give you a little bit of sample size for this 163. We've got over almost 1 million loci, and here's our statistics. We put our look at our original loci, we put in climate variables, and out we come up with the loci correlated with climate variables. So there's the assumption that when you get a correlation that they're going to actually give you a map, and then you get these adaptive loci. And just to cut to the chase, this is the map, what we call a map of maladaptedness. Dark red means it's not very, very good relative to the lighter colors. And also always remember with landscape genomics, it's just relative to each other. So at some point you've got to ground truth that. And so here's a lot of areas. You may not want to lose all of those areas. So how you manage it to get it so that they're not going to be jeopardized is a really serious question. So, so we've got that's just what landscape genomics tells us with blue oak. I'm trying to give you lots of examples, so I'm just going to move on to island oak, which is another project we did with some other collaborators at the Nature Conservancy. And here's what it looks like. If you looked at the Channel Islands for island oak, you see Catalina is far more maladapted relative to what we see in Santa Rosa and Santa Cruz. And in fact, Santa Rosa would be a really great place. So these are kind of, initial patterns. I'm going to just, I realize many people are following this. I can't go backwards. Okay. So I've given you just 3 quick studies that show you maladaptation. I'm studying about 8 species of oaks, and I could show you other oaks that are maladapted. Many of you have been showing it in Mexico. So I think I've established oaks are in jeopardy. And when we think about restoring populations and planting things, we need to take into account, are what we plant and put in the ground going to survive in 50 years? And that is my concern. So what are some management strategies? So I'm going to show you how we used our data for the oaks to look at, well, what could we do? So what we did is we looked at case number 1, seed transfer strategies for Valley Oak. Now this is based on phenotypic data, which I think is the most reliable. And so what we did here is we had 4 categories of how we're going to select. And we're just going to say, does it make a difference if we look at how they grow in the common garden and what trees we pick to plant? So we had one where we just said, what if we just randomly selected seeds? What if we did is we selected seeds based on the maternal site temperature with the cooler garden? What if we select seeds and matching the maternal temperature with the warden garden? And what if we selected the top performance from the cooler garden? So what we're going to do is actually look at the performance in the cooler garden and predict how they're going to grow in the warmer garden, not too different than what climate change is going to do. And you look at the comparison, and the main thing I want to show, okay, is that this group where you planted the best growers, you are going to get much better results. So if you look in your common garden, in your environment, and this is when we looked at who was growing well in a cooler garden, and we said, what would happen? How are they growing? How are they growing in the warmer garden? And we see a significantly higher result. So the implication is seed zones that people are now using as common practice may not be as good if you had phenotypic data as just looking at who's growing well. Case 2 is we looked at the Provenance study and we combined it with genomic data. And in here, I'm going to just skip to the pictures of if you were to use, all right, someone help me. Okay. If you were going to use data where you combine phenotypic with genomic data, you ignored it, this is the current. But if you were to use seeds based on that common data, this is what you could do. You could completely get green with existing genetic variation. You could have a lot better growing trees. So, and this is the ideal. So there is actually genetic variation in our oak population sufficient, if we were to identify it, that we could actually create healthier forests. And then I want to just go to the third case study and talk about blue oak. We're only using landscape genetics, not the combined approach. And here, what we did is we compared, we had sites that we knew were not very good. This is for that blue oak. And then we had sites that we knew would actually be really good. So think about those red sites versus light. And then we compared randomly selected trees that are close to the restoration site so you don't mess up the genetic structure. We did seeds that are adapted to the climate of the restoration, and then we just look at seeds that we are predicting to be highly adapted in the future. And in the low, so that latter category of picking seeds adapted to the future do a lot better than all of those other tests. And when you have if you have a site that's a really good site, we already know it's a very adapted site, you're going to get more. So, summary of the tests, basically based on the Provenance study, we can show that there are strategies that we can use that would be very helpful in managing oak populations. I showed it again with Labata when I combined phenotype and genetic information. And then if we use landscape genomics with the assumption that you have, you can also do a better job of deciding where to get the genotypes from to plant your forests. So in California, planting forests is quite common because they burn down a lot and we're restoring constantly and planting constantly. So I'm trying to do is think about getting the people who are doing the restoration to plant genotypes that are going to do well. It's expensive to restore a site. So to restore a site with bad genotypes, to me, is really, really a very, very sad exercise. So I have these recommendations. In the ideal world, if I had lots of money, I would combine landscape genomic data before I would start my restoration. I would identify the seed sources. And you might also want to, if you have to purchase lands with low adaptedness, it's going to be even more important to manage them well. But I would also, if you base it solely, I would do a parallel seed, kind of a common garden selective seedling experiment. Because what we're finding is in the first 3 years of seedling growth, it's actually predicting how they're going to do it 10 years. So I would say, let's do adaptive management and test our work as we go. If resources were limited, I would do seedling experiments and pick the best growers. I would still, so there's some low cost ways. Okay, closing reflections. I went through this very quickly, and at the end of the day, I have to thank you for your patience. One is, okay, increased fires, pathogen outbreaks, introduced pests, invasive species, climate warming, and landscape transformation. Obviously, those are serious threats to oaks. So I'm really just dealing with one part. And we do, if we want to preserve areas, we have to think about all those problems. And we've been hearing talks about it. You know, landscape transformation and fragmentation is reducing the size of the oak, reducing genetic variation. This is a problem. So thinking about where to preserve things is important. But oaks are an important part of the solution for climate warming. Not only are they foundational species, the ecosystem, but they do generate you know, and mitigate the climate change. So losing more and more of our forest cover and having trees that are not growing optimally is not going to be helping us in the future. So management of oak populations, restoration is urgently needed. Common garden trials and landscape genomic studies are part of the solution. And, you know, we spend billions on human health. Let's spend billions on environmental health. And we've really got to change our, you know, our world, because eventually, if not already, environmental health is directly related to human health. So I suggested 2 methods of what you can do. And I just keep pointing out that if we do all these other things, but when we restore, we don't plant the right genotypes, we're sort of dooming ourselves. But the good news is, and I'm such an optimist, why else would I be trying to figure out what we can solve in a great crisis, is that oaks have so much genetic variation that with careful management of what we replant and how we manage our forests, but it's going to take a lot of human intervention, but we could do it. And a lot of problems don't actually have a solution. So with that, thank you very much.
Thank you so much, Victoria. It's great to finally hear a talk from you after so many Zoom appearances. Does anyone have a question for Victoria? Iram?
Try in English or what do you expect about the restoration? Is possible to change for the pines with the ponderosa pines is maybe lost right now or is almost in this way, and change for the forest with oaks that is very more robust for this chain of forest?
You know, you really do ask a good question that obviously I did not address. There is some shifting of species distributions. There's not much we can do about. You know, so whether when you lose the pines, are you wanting to help and move the oaks up? I focus mostly on how can we keep the ecosystems that we have. Now, if we want to start shifting and bringing things up, that's going to take another kind of intervention. And whether or not we want to keep pines, and it's even possible to keep pines, that would be, I haven't looked at the data on that. That's actually an interesting question. What if, because they do a lot of pine planting of forests, right? What if they were looking at pines and wanting to keep them? There's probably some areas that we're going to have to really think about whether we keep those or we just allow the system. But in terms of the time frame, we need to move. If we're going to be moving oaks up and helping them, it's not going to happen by just waiting for natural migration. So that's again going to be a very active intervention that's going to be needed.
I think we can do one more question.
Thanks. Thank you, Victoria. You were talking about combining phenotypic information with genetic information, but how do you think it would be the better way to do this? Because, for example, there's also epigenetic variation, which you have studied, and also now with whole genome sequencing, we can access structural variants that are also important for adaptation. So how could we combine all that, to find like the optimal?
What are you trying to set me up? I have another part of my research program that, of course, is looking at these. I am looking at the molecular level, trying to see is there other, ways that we could be engineering or finding like, well, what kind of epigenetic response? I'm hoping to do I have a research project now to see to what extent does drought stress change the methylation, the phenotypes, the response to drought? Does it actually hurt the ability of plants physiologically to respond? Or does it trigger something that turns them on sooner? We don't know about these kind of short-term stress. So yeah, that's another part of the problem. And I sort of see this as what we can do right now and make a difference. And I see us as having to do a lot more on the biology of response to stress to see how much that's actually going to moderate it.
All right, thank you so much, Victoria. Okay, last but not least, in our session this evening, we have Zarah Wyly, who comes from the Land Conservancy of San Luis Obispo County in California, USA. Zarah is an ecologist and certified arborist working on California's Central Coast. With past experience in oak woodland restoration, community education, managing acorn harvesting projects, and growing oaks for restoration, Zarah has recently shifted her focus to guiding the next generation of conservationists. She is the program manager of Learning Among the Oaks and works to ensure that young people develop a love of oaks and the places they live. And each year she takes about 1,800 local students outside to learn among the oaks. So now she'll share more about this work with us.
Great, thank you. And boy, what a hard act to follow. And end of the day, thank you everyone who is here. I know you're here for the picture, but hopefully you'll hang out with me for just a few minutes, and I'll show you a bunch of pictures of some adorable children that will hopefully lift your spirits. So for me, I met oak trees when I was pretty young. This is actually a tree I used to play in as a kid with some of my friends. We'd have little tea parties in here. And this is a big Quercus chrysolepis in Santa Cruz, California. So you've all been sitting for a long time. If you want, take a second, stand up, take a deep breath, stretch like an oak tree. Yeah, thank those oak trees for oxygen that they're providing for all of us to be here. And as you sit down, I want you to take a second and think about when you first met oak trees or when you first fell in love with them, because we've talked a lot about how wonderful they are. So how many of you were a kid when you first met or fell in love with oak trees? Oh, interesting. Okay. We're like half and half. Excellent. Wonderful. So I agree that getting our kids out into nature is really important because many of you, that's where your timeline started with all this crazy oakness. And look at the power of oak trees to bring all of us together from 20 different countries. That's pretty amazing to me. So these days, I am lucky enough to steward a program called Learning Among the Oaks. And this was started 20 years ago in San Luis Obispo County in a little tiny town called Santa Margarita. And it is surrounded by a ranch that is full of beautiful oak trees. So there was a teacher at the school and they thought, you know, why are we inside? There's this beautiful ranch with beautiful giant, this is a Quercus douglasii, beautiful oak trees. Can we go outside? Because we're learning about science and you can do that anywhere, especially here among the oaks. So that is what we do. We're kind of an unusual education program in that we are embedded in a land conservancy or a land trust. And the organization that I work for currently oversees and stewards 66,000 acres within San Luis Obispo County, much of which is various types of oak woodlands. And I am embedded there in this land trust because they realize that in order to have conservation in the future, we need people to love and respect the oaks so that we have conservation as we move through the generations. So Learning Among the Oaks is focused on elementary school children in public school. We actually go to where the kids are, and we have different programs for each grade level. Unfortunately, in the United States, you can't just be like, let's take the kids outside. There's all of these requirements to meet standards as far as public education. So all of our programs are aligned with next generation science standards and are focused on age and grade level appropriate topics. So we start off in transitional kindergarten. This is kind of a strange thing. It's like before kindergarten, we talk about if things are living or non-living. And we go all the way through to 5th grade, in which we talk about the carbon cycle. And Oak Woodlands is a perfect place to talk about carbon. Let's see. Every year we take about 1,800 students out to these programs. Most of them were able to actually take into woodlands adjacent to their schools, either onto private ranch lands. This photo is from a school that actually has their very own oak nature trail, which is pretty magical. Or sometimes we have to bus them to a public open space where they can hike and learn and explore. One thing that is really different about our program is it is very, very hands-on. How many people here really do not like snakes? Anyone? Oh, I see a couple. Okay, there's no shame, no fear. But as a hands-on program, one of the things we do is ensure that students have the ability to experience things that might be scary or new to them in a safe space. So for our 4th grade students, where we are starting to take them for longer and longer hikes, we always go into the classroom with some educational snake ambassadors that we borrow from a local university and allow students to learn about safe snakes and also unsafe snakes, because we do live in an area with western rattlesnakes and we find them on almost every single hike we do with kids. So it's an opportunity for people to realize that snakes are not scary, but it is a way that students can then go out with their families, feel comfortable, know how to be safe in snake country, and then maybe even find a little bit of joy in meeting snakes. This little boy was really funny. His parents are farm workers, and they have some very negative experiences with rattlesnakes in the fields where you harvest grapes. Often rattlesnakes will end up in the grape hoppers when they're using mechanical harvesting. And so this young man had only heard that snakes are bad. So this day, when he got to meet, this is Milo, the gopher snake, he had a great time, and we sent this picture to his parents and had a wonderful conversation. So another thing that is really unusual about our program is youth leadership. We try as the adults to do as little talking as possible. So we get the fifth graders, the older students, to do all of our work for us. So when they're in fourth grade, they can apply to be leaders, and we call them Oak Ambassadors. And they get this special training in which they learn all of the topics and have the opportunity to gain mastery in these topics by teaching them to younger students. And it depends on which grade level we're talking about. Here, our Oak Ambassadors are getting ready to do a puppet show all about habitats. And in this one, all of these creatures end up in the same hollow log because it has great habitat features for all of these animals. After they graduate from elementary school, which is usually fifth grade where we're at, the students are given the opportunity to continue with us as Oak Ambassadors throughout middle and high school until they become adults. And this hands-on opportunity continues. In this picture, I'm showing one of our Oak Ambassadors who designed this whole cool contraption. This is a small animal camera trap. We also have a large animal camera, wildlife camera system that the students help manage. But on one of our preserve properties, we had questions about what small animals were actually present. We had some questions about if the list of animals was actually accurate. And so we said, hey, that's a great opportunity for one of our older Oak Ambassadors. So she designed this camera trap, and there's a little fence there. So the animals come down as they're moving across the landscape, and this is right at a transition zone between an open grassland and our little riparian area. And they are forced into this box where they get their glamour shot. We take lots of pictures, and then they continue on their way. And so she built this contraption, she tested it out, and also gathered data for us at 3 months in 2 different locations, and was able to actually capture pictures of a bunch of small species that we had never seen before. Along with this project, we also have opportunities to manage Western bluebird boxes. We need to provide more habitat for them. They've been in decline in our state because of the loss of older oak woodlands and cavities. They're secondary cavity nesters. And this last year, our students were able to record 70 new baby bluebird chicks coming out of their boxes during the season. They also do help with things like sudden oak death monitoring, and they also help maintain a couple habitat gardens that we have on school campuses. So there's really a lot of science behind environmental education, and that it is really valuable. There's studies that show even 30 years later, there is still an impact from quality environmental education being received during elementary school. And so that's why we're really focused on starting with students as young as transitional kindergarten. These students are looking for, I think, flowers on their little scavenger hunt here. And being able to do successive lessons with these students throughout the years, it really is also becomes kind of place-based learning. They learn about what they have locally. And, you know, instead of learning about all these animals in Africa or in Mexico or all these cool foreign places, they get to start at home and see what kind of bugs are in the flower box outside their classroom. Yeah, we'll move on. So I kind of see this a little bit as rewilding the children. I don't know about all of you, but I spent a lot of time outside as a kid. And I saw a recent paper that said young people spend 50% less time outside than their parents did, which makes me really, really sad. Another study showed that young people, at least in the United States, they spend as little as 4 minutes a day outside in unstructured play. So not accounting for things like going to soccer practice, kids spend very, very little time outside being curious. and learning about the world. So I'm going to just flip through a bunch of pictures of our kids having a great time. We're lucky to also have a nature classroom in one of the schools. They had an extra room, so we filled it with all sorts of fun things that people can touch and learn about and explore, because hands-on is the way to do it. I mentioned this briefly before, but with our younger students, we keep them on their school classrooms because we really want to make sure that everyone understands that nature is a thing that we're a part of and is everywhere. It's not something you just go visit. And our older students, they really do like to get outside and visit, and we make sure that everything is activity-based. These students are playing a game in which they have to pass acorns with their fingers like various types of bird feet, and some of it is very, very challenging and lots of fun. So joy is definitely a big part of every program we do. And again, having the older students to be role models for the younger students is really impactful for our program. We usually let them maintain the rules here, which include things like making safe choices, using quiet voices, and not taking things from the forest. And they learn a whole lot of really wonderful skills too. And curiosity and joy are really important. This boy here on the right, I don't know if you can see what this little crazy animal is. This is a legless lizard. And this is a second grader who found it during a bio blitz and was over the moon the rest of the day about the thing that he found. So even though we have these very strict curriculum that we're following, we make sure that if there's an opportunity for joy, we follow that and follow the kids' lead. And sometimes we actually don't get any of our curriculum done, but don't tell the teachers. And here's our fifth graders. They're actually measuring the size of this tree using arborist tools so they can calculate how much carbon is in their school oak woodland. So doing things that actually have an impact and actually are related to things that they might do in the future is really important. As is making friends. We often forget the social emotional part of being a kid and being outside, but we often see students that don't necessarily spend time together in the classroom, forming bonds and alliances when they're outside. And just being happy. I would much rather be outside than in a classroom, and I think 100% of the students that we interact with agree. I need to make sure to talk about two important things, which is why I put this slide here. We have a lot of partners. On the left, you'll see there's a Virginia opossum that was brought into this class so that they could learn about this important native mammal. This is from a local wildlife rescue group. They have animals that cannot be rewilded, and they share them with our students. And on the right side, I want to point out this is a really cool sheet about oak galls that we use. All of our printed materials are in both English and Espanol because our entire program is bilingual. We found that over 50% of the students in our program speak Spanish at home. And so this year we made the effort to convert all of our educational materials. And also our newest educator that we hired is also bilingual. And that has been really fun for all of our team and all of the students. To see a kindergartner who is in school listening to English all day, and that's not the language that they understand, to watch them perk up and get excited when the educator does her welcome and shares in multiple languages what we're doing that day is really fantastic. And instead of teaching them very specific things about nature, we spend a lot of time observing and asking questions. We really like to follow the nature journaling model of open-ended questions. We ask them, what did you notice? What did that remind you of? And on and on and on and on, because there's no wrong answers in our programs. There's only curiosity, excitement, and joy. And just some more kids being cute. I wanted to make sure I had lots of pictures for you. Lastly, but not least, I wanted to talk about inclusivity. We're lucky enough to have partners that provide us with things such as we have this really cool contraption that is an off-road wheelchair. So we're able to have anyone who has a mobility issue come join us on a hike. We also do provide interpreters for students that use sign language. And if necessary, we also are able to hire other language interpreters. So we're very excited to ensure that every student in our public school can come on our trip no matter what is happening. So I'm definitely not alone in this environmental education thing. I don't know if you noticed how many times today people spoke about this during their talks. I also noticed there's 3 posters out there on projects that have an environmental education component. So I'd like to, little clap for all of you for including that. You'll see there's other programs in California I know about, at least 4 that I've heard of here in Mexico. And I also want to point out that I am in contact with people in Granada, Spain with Operación Encina. They are doing youth education as part of their reforestation. And apparently there's also a group in Greece that is doing similar work. And if any of you know of more, I'd love to know about them because it really is a universal thing that brings us all together. And I'm very hopeful for the kids in the future because what people love, they'll protect. And I have a lot of hope around that. So thank you for your time. And for staying this late in the day, you deserve a round of applause.
Thank you so much, Sarah. That was incredible. Does anyone have a question?
Gracias. I'm indigenous to this place. I'm coastal Chumash. Haku Haku Shumawish Tipa Shumawish Lena Hilkanta. I'm just wondering, this is a beautiful program and I'm grateful that you're there doing it, but I'm just wondering what the local indigenous involvement is. I didn't hear you talk about any aspects of like through a Chumash lens or anything like that.
Yeah, that's a wonderful question. 3rd grade within the science standards is our place to talk about landscape change over time. We work very closely with Yaktitu Titu Talini, and they have looked at our program. And since we're science focused, we don't go really deeply into anything more than the long-term relationship of the indigenous people to these oak woodlands, their care and how that has changed the landscape over time. We also are very cognizant of the fact that this oak community is still being stewarded. And so the land trust that I work for has a really deep relationship with all of our local tribal groups. And I'm excited to see you here. So welcome. Thank you.
Can I?
Oh,
yeah.
Go ahead.
So wonderful program. Wonderful program. How many days a year do you get to spend with the students? And are you in the classroom or are you an after school program?
Yeah, that's a good question. For me, unfortunately, I'm only there a couple days a week because I have to do all the program coordination and make sure we still have funding. But we are at a school every single day of the year doing some sort of programming. We are not currently doing after school programming, but there's a lot of requests for that space to have people come work in it. There's actually almost more funding for after school programs. So if anyone is wanting to add local oak education, I would absolutely look at after school programs.
So a given student will be getting a science class in your oak curriculum twice a week. How many times? How many times does a student experience this curriculum?
Yeah, so that's a good question. So we're at 11 different elementary schools, and we're a team of 4 people. So we see every elementary school student at least twice a year. We see our 5th grade Oak ambassadors once a week. So it's really as they get older that we get to see them more regularly. But with the number of staff, that is as much capacity as we can handle as far as being at a school, because we go into a classroom before we do an outdoor lesson, and then we do an outdoor lesson. And many of our schools have 5 to 7 classes for each grade level. So it's really a huge time commitment.
Any other questions? I haven't gotten to ask one yet during the session, and I, kind of similar to Victoria, was wondering, I guess apart from the schools that you regularly visit and give programming to, are there any that just come to you all for maybe like a session one time during the year when they want to get the kids outside?
Yeah, absolutely. So we're fully embedded in 3 schools, and then we bring in another 8 schools for one-time hikes for various grade levels, which is very confusing. And I should share that all of this is, we do not charge the schools for this funding. And as far as the time that we have available, we have an equity schedule as far as which students we have to provide funding when we have another day and can sneak a class in.
Okay, well, thank you so much, Sarah. And I guess another round of applause for all of the speakers. I can do it. I see on the program that next is the group photo, which is nice because the room looks full again. So
Okay, so the group photo is a very important part of our conference. We have a long tradition having these great photos that we can keep as a record of who is here. So I would encourage everyone, please, if you register for the conference, let's do this photo. We're going to do it by the swimming pool. So on the way down, when you went down to the lunch, you saw the swimming pool, you turn right. So go down. It has some wonderful steps, layered steps we can all form up on. So please go immediately. Do not delay so we can get this done quickly. Lo voy a decir en español también. Es muy importante para nosotros tener un registro de todos quienes estuvieron en esta conferencia. Por favor, vayan ahora a la alberca. Ahí vamos a sacar la foto. Fórmense en los escalones que hay en la alberca, por favor. Y luego, after the photo,